Hey, good morning. Let's get started. 9:15. All right. A very good morning to you. Welcome to this rather marvelous reading room here of the Welch Medical School Library in East Baltimore. Welcome to the 2022 IDEAS Annual Symposium. For those of you who don't know me, I'm Charles Meneveau. I serve as the Associate Director of IDEAS. I've been for a while. I'm a professor in mechanical engineering. My area is turbulence and big data. And so welcome here. Alex Saleh, our director, would have been the one to welcome you and host this. Due to concerns about immunocompromised conditions, he preferred today to stay to participate remotely. So he's on the Zoom, but because this is our first time in person again since a few years, I guess, we thought it'd be good. He asked me to welcome you in person. Welcome to the symposium in Alex's name. As is usual, I'll give you around 15 minutes or so of introductions about IDEAS. And since I know there's quite a few new members, new people to the Institute, I thought I'd start with some general descriptions of IDEAS. Who we are. IDEAS is a team of researchers and engineers actively helping research groups for best solutions to meet their data needs and fill gaps and broad variety of technical expertise and experience. This is what you'll find on the IDEAS website, and I think it does describe us quite well. Quite impressively, it's a very it really covers pretty much most of Johns Hopkins, Krieger School, Whiting, the libraries. Those were the three initial parts of the university that founded IDEAS, and then the Keyi Business School, School of Medicine, Public Health, of course, great additions. Approximate numbers, there's over 150 members, over 20 technical and support staff. There are a number of affiliate centers and other institutes of the university that are sort of enmeshed with IDEAS. There's an IDEAS data center. We have the SCI server that you'll hear about today, and Arch, the successor of Archie that you'll also hear about. And essentially, the vision is that we lead the translation of data science and technology to real-world problems across the university. There's an executive committee. Alex is the director. Then there's essentially 11 additional people covering the various schools and various aspects of IDEAS. We meet once a month to discuss all sorts of things. Let me tell you a little bit about the departures and arrivals. Saeed Choudhury was one of the original executive committee members. He was representing the library. It was had quite a lot of vision and influenced IDEAS a lot, as well as the synergies with the library. He has moved on, and Tara Engel, up to last year, was the very able IDEAS administrator. So we thank them both for excellent, outstanding service. A number of new people have joined. Peter Lawson, whom you hear about today, we'll talk about the library, so he's representing the library, the executive committee member the executive committee. Elliot Rosen, new administrator, so he has a lot to do with all the organization today. Kate Stevens as well, our communication specialist. She also has a lot to do about the organization. And then a number of additional people have joined IDEAS, so welcome to everybody. And then Jeff Leake, a long-term collaborator, has also left, and we thank him for all the great contributions over the years. Now, what we do here, I'd like to cite something that Alex described, I think, a few years ago. So we do convergent open science with interactive petabytes. I really like this. So we provide disruptive assistance, and the idea is to institutionalize across institution lessons learned in this multidisciplinary setting. But one thing that's really clear is that one size fits all is false. We do not subscribe to that, and there's multiple levels of both science engagements across their various phases of maturity, and then levels of engagements, the type of people are involved, architects, disruptors, and implementers. And there's essentially also a critical mass of postdocs and programmers that help everybody. So again, from the website, there's the services, the sort of consulting services that IDEAS provides in terms of developer, programmer, and systems admin consulting. There is a number of convergent multidisciplinary engagements across many different disciplines: astronomy, oceans, turbulence, material science, computers, bioinformatics, social sciences, city planning, climate modeling, and cancer imaging, and so on. There's collaborations with national labs, federal agencies. There's educational initiatives and outreach programs. And essentially, the idea is to leverage our scalable open infrastructure. Another thing we do is we host and maintain some of the largest world-renowned scientific databases. There's a number of examples, again, starting with the astronomy and Recount2, turbulence, ocean circulation, and many others. Their IDEAS has a grant submission service. So again, those of you writing proposals to agencies, you can avail yourself of this service through IDEAS. Then again, something you'll hear about today, there's seed grants and fellowships that IDEAS awards competitively. It's very interesting things happening. And then of course, events such as this one. There's a big IDEAS seminar series every so often. We bring external speakers, there's workshops and so on and so forth. Flagship projects that we thought might be, so one of the important ones, most sort of will last or have lasted for quite a while. There's SciServer. So this is a platform that you will hear about again later today, which is a scalable data aggregator for a number of fields. It's the official science platform for the Sloan Digital Sky Survey. Copies of it have been deployed in various places. School of Medicine, there's a copy, there's in Japan and Max Planck in Germany, as well as NIST, that we heard last year from an invited speaker last year about the implementation there. It also is the platform that hosts part of the turbulence and the ocean circulation data. And then something new, the AstroPath 2021 is essentially an astronomical image analysis meets cell pathology type of project. And it's been essentially an award-winning collaboration with the School of Medicine. Spatial transcriptomics, it's making already a major impact on cancer immunotherapies, and essentially also building data sets that can serve as ground truth to train machine learning. New projects and directions. So there's the National Science Data Fabric, new turbulence project that was awarded last year, and then new collaborations with Hemi, NIST, I already mentioned, the ROSI, there's a wind farm database project that's going to be a poster today, and other international partners in academia and industry. Now, let me just very briefly, and we'll hear from KT Ramesh about this, sustainability and future of IDEAS. IDEAS will be part of AIX. This is, as you will hear, a new pan-institutional center to advance artificial intelligence technologies. This is quite a big thing for the university, and we believe that the presence of IDEAS on campus for a number of years already really did help facilitate and give gravitas to AI and computing on campus. I believe it's made this has helped facilitate the creation of AIX, and so IDEAS will play a role in its specific subfield as part of AIX. And then, of course, very important, and Gerard Lemson, following me, will say a few words about the Scientific Software Engineering Center that's hosted within AIX already, which consists of a core group of data scientists and software engineers coming to campus. So about this, Alex likes to close his talks with little sayings. So I believe last year he talked about this, about leadership. Those who walk in other tracks leave no footprint. And, with this connection now with AIX, I believe this is true. However, if you have a bigger shoe, then this may not be necessarily true. So I think the growth in this direction really is making a lot of things possible. Another thing maybe that I mentioned is about IDEAS itself, in terms of how, there's some people say IDIES, other people say IDEAS. That's not really what I want to talk about. I just want to mention that working with computer science people and so on, there are some sayings and some words that I find fascinating. There's the wonderful words such as embarrassingly parallel. These kind of juxtaposition of words I love. I think Alex likes another one, unfair advantage. These are great. So when I think of IDEAS, I think of insanely diverse. And so maybe for today, we'll think of IDEAS as insanely diverse intellectually and entirely seriously. Again, I think you'll see in terms of intellectual diversity, you'll see a lot of examples of this throughout the day. So welcome to the symposium and enjoy the day. Thank you. All right, and so I am going to chair the this first session this morning. So I'm going to introduce Gerard Lemson. Many of you know Gerard. He's going to talk about this new institute. Let's see where the things. Where it is.
Yes, hi. Charles already mentioned this. This year we got money from Schmidt Futures Foundation, started by Eric Schmidt, the former CEO of Google. And in particular, it's in the context of what they call a Virtual Institute for Scientific Software, VISS. So what they try to do is they try to accelerate pace, as I say, of scientific discovery by supporting scientists with money, and in our case also with developers and engineers. So they want to, as I say, address the growing demand for actual professional software developers. If scientists have a project, they don't Schmidt thinks that it would be good if they get, say, professional software developers, rather than rely on graduate students, postdocs, etc., who may go away or don't have the background, who have to write papers, etc. So to do this, what they've done is they've started in this first phase 4 SSECs. You don't get that's a bit of an ugly acronym. Some people call it SSEC or whatever it is. So there's one in Georgia Tech, one in Cambridge, University of Cambridge in the UK, one in University of Washington in Seattle, and there's one at JSU. So these will be run by these bigger universities. They will work within the university and work together to come up and assign projects from people who make their expression of interest. I mean, we're currently in the phase of how to define these things, and they get assigned people who are going to work in these institutes. So we are one of them. So we started, I think, formally in March. I'll tell you a little bit about what we're actually trying to do. We've had some activities already. The website is still a bit in process. As you see, it is under the AIX flag, under the AIX umbrella, ai.jhu.edu. So the current organization is basically, there's a team at IDEAS. It's Alex as director. I'm the associate director, and Kate Stevens there is our communications specialist. We're going to hire people. So one of the things that Schmidt thinks is we need to have people with industry experience, with really the attitude to do software development right, to produce tools that can live for a long time, platforms, etc. So we have to hire people, and they give us money. I'll talk about that later. Some of the issues that are coming in there are somewhat disruptive, you could argue. So it has been, the SSEC has been set up under the AIX umbrella. So Katie, Ramesh, and Alex worked on this together. And we get a lot of help from Scott McGee and Katie Vaught from their side. but it is embedded within IDEAS, meaning that if you go to the 6th floor of the Bloomberg building on the Homewood campus, the last 4 offices will be dedicated to people from the SSEC. So they will work together. They will, well, work together, yes, but they will definitely be embedded within IDEAS, and hopefully there's a lot of cross-fertilization there. Okay, within this VIS, we are focusing a lot on data-intensive science. The University of Cambridge, they basically have a specialization on climate modeling. Georgia Tech has probably more library development. University of Washington is a bit similar to us. Actually, they have an eScience Institute, which is probably bigger than IDEAS. That has been there for a while. But so what we, as Alex always said, tries to do is we want to try to give people interactive access to petabyte data sets. So we have expertise already from IDEAS, and we would like to leverage that in the new projects. In a sense, the things that the IDEAS have been doing, which is working generally the scientists within Johns Hopkins and a bit above, is what the SSEC will do, but there the projects will come from the whole country, maybe from abroad as well. So the thing is, we want to be able to compute everywhere. That's part of this NSDF project, and the goal is to start embedding AI on top of the data sets. So I'll say a little bit. So we've started only in March. We start the process of hiring people, but we've done 2 specific activities already. Some of you were involved in this or have been taking part. So the beginning of June, we had a workshop. I'll say a bit more about that. So there was a workshop for large-scale scientific imaging, June 16, 17. Many of you, I think, were there, but maybe not everybody. So motivation was that large-scale imaging is something that almost all disciplines work with. AI works, as we know, a lot with images. But on scientific images, we don't know that well what to do. It's hard to manage them, to administer them, to store them often. But almost all disciplines work with them. SDSS, which was basically the start of the IDEAS project, started with lots of imaging, like surveying the sky, but lots of other data sets have been come along since then. We also make images, basically synthetic images from simulations. We have loads of those already at IDEAS. And as we actually started seeing during the workshop, it's easy to start providing or to producing petabytes per year from all kinds of different fields. The problem is that most of the time the handling is done one thing at a time. And so the idea here was, can we do something more with that? So the goal of this was to bring people from lots of different disciplines together to have talks, panel discussions. This is accepted for technical reasons. We couldn't actually go there, but this is the website where you can get the slides, or the movies, the videos. Oh, according to Kate, it's working now. Good. So the goal was to bring together people from different disciplines and talk about all these problems. But in particular also to see, can we find commonalities? Can we find standard ways of doing things? Can we learn from each other, get synergies? And also one of the what Alex wanted to know from people, if you had unlimited storage, compute, money, what would you really like to do? And there was a panel discussion, a couple of panel discussions afterwards, which are not going to be made public because we want to find out, but can we come up with a couple of blue sky projects for which we could maybe ask the university or funding to come up with, to actually come up with some of the, well, the most advanced ways of handling images so far. So these were all the talks. I won't show you, I won't go into them. But so there were talks focused on microscopy, astronomy, SDSS, but also from UW, Andy Connolly, who was one of the directors of their SSEC. Material science, people here in the room, people doing simulations, lots of talks on medicine, obviously. These are all already supported by IDEAS. So IDEAS is, as Charles already said, very cross-disciplinary. They're not necessarily all supported in the same way, but since we have them all already, many of them in our data center, some nearby, some of them using the SciServer platform, we basically have a lot of this expertise. And so but it's actually nice to see this. Okay, so that was the workshop. The panel discussions were very interesting. As I said, there are videos of everything. There are videos of the presentations that are publicly online if you want to follow them. The other ones we're going to dig in and see what conclusions can we draw from them. So another thing that we had planned already, since SSEC is embedded with IDEIES, and since IDEIES already has developers that are similar to the kind of developers that the SSEC is supposed to hire, we thought, are there already some projects available provided by Schmidt Futures that some of our people could work with, so we can get experience with that. We can give feedback back to the Schmidt Futures leadership. And so there was they offered us 3 projects. One of them basically fits all the boxes for us that with the experience that we have already. It's called Democratizing Data. It's led by Julia Lane and Nancy Potok. Julia is a professor at NYU Wagner. Nancy is CEO of NAPEX Consulting, but both of them had actually expertise. They worked with the White House. Nancy was formerly the chief statistician of the country or something like that. So what they want to do is they talk to especially federal agencies who are interested in this. The federal agencies collect large data sets, make them public to the world, but they would also like to see how much of that data is actually used. How valuable are the data sets? So how do you find this out? And so the goal was to go to publications. In this case, they talked to Elsevier, there's lots of publications, and start searching, developing tools to search the publications to find out references to data sets. Because it would be great if everybody was using DOIs, digital object identifiers, pointing to publications already in their papers, but that's not the case. So what they want to see is which publications are used which data sets are used in publications, maybe to find out which data sets are used in conjunction, which people are experts on this, etc. So basically, they created some tools, first using a Calgo competition. Part of the project is to develop those further. There's also sponsors. So Schmidt Futures sponsored the initial part, the proof of the prototype phase, and there's other ones as well. So our role is first that this was the kind of a proof of concept project with people that we already have that are similar to the people that the SSEC will want to hire. Maybe those must have industry experience, who knows? But so we, with Eric, Mitya, and Manu, sorry Manu for not putting your last name there, it's too late. We started working. Julia and Nancy are very happy with it. We're now transitioning over. They were paid originally from SSEC money, but money is coming in already from different funding. So the different funding sources from McGovern and others. Every agency starts supplying money and we get part of that. So Juliana and Ryan are brought on board for UI development and more data science things. But so our goal is basically support the core workflow of the whole project. They had in a proof of concept something running on somebody's desktop at NYU. It's now running inside a SciServer. So there's an ingestion pipeline. I won't go into details of it. Elsevier produces metadata about publications. We wrangle it, put it in the data model, make it available for validation by actual members of the agencies to say, yes, this reference that you think is pointing to a data set is indeed that particular data set or not. This is going to be fed back, etc. Then it's moved on to TAC, the Texas Austin Computing Center, Advanced Computing Center, sorry, what the A stands for. And they provide APIs, dashboards, etc. So we work on a data model, don't worry about that. The validation tool is a tool that basically, when people log in, they get a snippet of text from a publication, and they have to say yes, no, unsure, whether it is a reference to a data set or to that specific one that the agency is interested in. So this is building almost like a training set, but this is the data that then finally the agencies are interested in. We've created, Manu has created nice dashboards within SciServer that then can be exported as standalone tools for visualizing. The graphs are basically connections between people in papers or data sets in papers. So we can go all the way. And we will also, because the data set is accessible within SciServer, server, people can start drilling down even further by writing the queries they want, doing whatever analysis they want themselves. Okay, so this is running. We're basically a very good, what is it, context. They like the way we do things. As I said, right now it is moved from SSEC to IDIES. We get separate funding, and it will keep on going for who knows how long. So basically, almost done. The first observations about the SSEC. We will have to hire people. And one of the things that Smith says, you want to hire people who are already in industry. There's a big gap between salaries in industry, salaries in science. What we would like is that it goes this. That will not like happen, but bring them a bit closer. And so how to do that, we'll have to see. What we would like anyway is people who have maybe a graduate, maybe a PhD already in science, and would like to come back to science, like some of us have done. You know, we've been in industry, coming back, and so you take maybe a salary cut, but we still have to see how that goes. Because, I mean, so the problem is, if you do this, how do we preserve equity? So AIX is also one of the initiatives that tries to work in that same direction. So on the other hand, it's really, so far, we have really liked the project. I mean, it promises a good. So if we get people who come from science, have a lot of experience there, some from industry, we learn a lot. I mean, in a sense, Eric taught us a lot coming from Amazon. No, and so you have this kind of an extra impact that we can all learn from. So that would be good. And these are all somewhat standard things that I have to say why we're happy and enthusiastic in the meeting we had at Schmidt Futures at some point. But so one of the important things for us is that SSEC can also be seen as a kind of a prototype for how AIX will start hiring people, or AIX Foundry. So we hope that it will be lead to a lot of cross-fertilization with IDIES itself. And the particular project that we got, we really like working on. Now, all of us were quite enthusiastic doing that and trying to work on that longer. Schmidt is probably going to say, okay, they are going to have these expressions of interest, and that project will be 3, maybe 9 months. Still have to start talking about the possible sustainability beyond that. But as an example, if all of them are like this, we would really like to go forward with it. So thank you.
We do have a little bit of time for one of the questions if anybody wants to ask questions about this new center. Yes.
So, I agree with you with the difference between salaries, academic industry. Even though we're doing better now, we're still not at that point. And that is a big point of but industry offers them stock options that we cannot offer at the time. And that is what the sticking point. In this approach that every industry has, it's not related to IDIES of the Hopkins, it's everything that is because of the stock options.
Yeah, and AIX, I mean, KT may say more about that. I mean, they're talking to HR to see what can be done there. You know, stock options, we don't have.
We offer academic credits. All right. Let's,
okay. Thank you.
Tom,
yeah.
I think the agents, my NSF, the federal agencies are keenly aware of a challenge to recruit and retain software scientists, software engineers as well. At least that's what the program managers have been telling me.
Yes.
So my question is, what's the strategy to engage with NSF or agencies?
I don't know if they have a particular strategy, like in this case, Schmidt Futures. The thing is, Schmidt Futures gets us for 5 years. Maybe they will extend it, but ultimately it will be different. No, it will, SSEC will be subsumed within AIX Foundry, and how that will, so that will be working for us. But I don't know where the funding, I mean, if the funding comes from internal and we can keep people, then hopefully NSF projects don't have to start asking for the same amounts of money. It will be almost like our own contribution. I don't know how it will work, but So I don't know if Sweet Futures themselves, they must be thinking about it, but I'm not aware yet of particular strategies there.
Excellent. And I think I'm really welcome.
All right. Well, thanks very much, Gerard. All right, so now we go to our first keynote speaker, Stephanie Hicks. Welcome, Stephanie. All right, so Stephanie Hicks is in the Department of Biostatistics. She's associate professor there in the Bloomberg School of Public Health. So she works at the intersection of data science and health, and is a faculty member of the Johns Hopkins Data Science Lab and the Center for Computational Biology. She's a co-host of the co-founder of the Our Ladies of Baltimore. So I saw that. So R being the famous statistics package. That's great. and also corresponding author of a podcast. So we're very pleased that she's giving us a keynote lecture today. The title is Scaling Up Single Cell and Spatial Data Analysis. And we need to find your slides, though. For that, I will. You know how to do this?
Yeah, I'm gonna go to my website.
Oh, you go to your website.
It's in the chat. Okay.
All right.
Okay,
Well,
I'll click that.
welcome, Stephanie.
Thank you. Okay, and then I'm gonna just hide this real fast. Oh, no. Hello, there it goes. Okay. Thank you, everyone. Thank you for the invitation to present today. I'm really excited to share with you some of the work that I've been doing in scaling up statistical methods and computational methods for the analysis of single cell and spatial transcriptomics data. Okay, that works. So if you are familiar with gene expression data, the way that you might have encountered them originally is in this concept of microarrays or bulk RNA sequencing. I ground us in this opening slide just to tell us that bulk RNA sequencing is an average of expression across all the cells in a tissue that you measure. So for example, if I take a tissue from a tumor or a brain, for each feature that I measure or for each gene that I measure, I just see an average of expression across all the cells in the tissue. An analogy I like to think about are Legos. Imagine you have a big pile of Legos. Bulk RNA-seq is the concept of maybe measuring the width of all the Legos and just seeing an average of widths across all those Legos. Fast forward to 2014, 2015, when single-cell transcriptomics came commercialized. This is when these technologies really took off. And the idea is now we get a full molecular readout for each observation or each cell. So now we capture maybe up to 20,000 human protein coding genes per cell. So really amplifying the sizes of the data, but also the resolution of the transcriptome that we get to see. So the analogy here is imagine you take the big pile of Legos and separate them into little piles by color or by shape. Now you get to see the different types of cells. Maybe you get to see a difference of relative abundance of each cell and so forth. So formally, single-cell RNA sequencing is the concept where you take a tissue, you isolate or disassociate each individual cell into little aliquots, and then we capture or measure the full molecular readout or transcriptome of each individual cell. It goes through a sequencing process. At the end of that sequencing process, you get a reads. That's what's shown there in the top right corner, those little bars. And we figure out how they map best to some reference transcriptome. In this case, I'm using the black box as gene 1 in our reference transcriptome. We count up the black bars, there are 18 of them, and we record those in a matrix. So in the row 1, column 1, we record 18 corresponding. There were 18 molecules expressed from this gene in the cell at a given time. This matrix is very sparse compared to bulk RNA sequencing. In bulk RNA sequencing, it's typically max, maybe 20% zeros, but in single cell, that matrix can be anywhere between 50 and 99% zeros, so very sparse. We can do a lot of things with these data. We can plot these data and see how the expression changes across different genes and different cells. We can apply dimensionality reduction techniques. We can take it from 20,000 features into a low dimensional space using such things as principal components analysis and so forth. Okay, so I start with a really great worldwide organization called the Human Cell Atlas. So the Human Cell Atlas has a goal of creating a comprehensive reference map for every cell in the human body. It's a really lofty goal. In 2018, they published a white paper saying that they had a goal of creating an initial draft of this map or this atlas of 10 billion observations, where each observation is an individual cell, and each cell can capture maybe up to 20,000 features. That's a large amount of data. And so you could ask, that's a pretty extreme example, Stephanie. What are examples of sizes of data sets today? So these are 4 papers from 2020, so 2 years ago. And I'm pulling out and showing you the number of observations that were sequenced in each one of these studies. So for example, you can see that we are on average at least sequencing around 2 million cells per data set. So each one of these studies that flies by, Twitter, for example, we are producing very large data sets. So one of the tasks or one of the goals that I'm interested in is thinking about how to scale up statistical methods or computational methods for the analysis of single cell data to millions or potentially billions of cells. I'm going to focus next on an example of one area or one goal in the analysis of single cell data, and that's clustering. So if we want to cluster millions of cells or billions of cells and identify how many different cell types there are in our data, we need a couple of things. One, we need to be able to work with data in memory or data on disk. Traditionally, in the world of single cell, everybody's used to working with data in memory. But as data sets become larger and larger, we need to be able to have computational methods that seamlessly work with data in memory or on disk. And there are many examples of on-disk data formats, such as the HDF5 file, which has been around a long time. There are some newer file formats, such as ZAR and TileDB, which are really exciting and have a lot of great potential. I point out that these are the file formats that the Human Cell Atlas and other large-scale organizations are using to store their single-cell data. So if you go to want to use these data, you will encounter these file formats. We also want these methods to be fast, so we don't want to wait around, a week to have a clustering algorithm get us a set of answers or predicted labels for each observation, which cell type it belongs to. We may want to be able to do this multiple times. So imagine we want to use K-means. So K-means is a well-known studied algorithm to be able, an unsupervised clustering algorithm to be able to group observations into K groups. Maybe we want to try clustering with K-means multiple times, try K equals 5, K equals 6, K equals 7, and so forth. This may be able to fit inside of memory if we apply something like principal components analysis, or maybe not, depending on if we want to use the full transcriptome. So for anybody who's not aware of K-means, one quick slide on K-means. So formally, K-means is, the idea is to take a set of n data points, maybe our 10 billion observations, call that X, and a number K. This is user defined. K-means formally partitions the data into K different clusters using a within, or minimizing a within cluster sum of squares criterion. They're shown in the middle, where X is the matrix and mu I represent the centroids for each of the K groups. In practice, one of the standard ways to go about this is to use an iterative algorithm called Lloyd's algorithm, where we iterate back and forth based on 2 steps. One is the assignment step and one is the update step. In the assignment step, we are given a set of centroids, maybe just a random set of centroids for our K groups, and we assign each observation to its closest centroid. And then using those new labels, we update the centroids for each cluster and we iterate back and forth in this fashion until convergence for those mu i's. So K-means is really great. It's widely used outside of the world of single cell. It has been for a really long time. Problems with K-means are, 1, you have to have all data stored in memory. 2, you have to store all the pairwise distances between each observation and its closest centroid. So if imagine you have 10 billion observations, that's a really big distance matrix. So there are some newer approaches, for example, mini batch K-means. The idea is very similar to the K-means, except At each iteration in the algorithm, you only bring in maybe 1% of the data into memory, or 5% of the data into memory. Or maybe it's an absolute number. Maybe it's 100 observations or 500 observations. That quantity is referred to as a mini batch. And the idea is you load in 1% of the data, you update your centroids, you throw out that 1% of the data from memory. You next load in the next 1% of data, update the centroids, throw that out of memory. And at the very end of the algorithm, you do a final assignment step with your final estimates of the centroids. It's a really cool algorithm that makes clustering incredibly scalable. Meaning we don't have to store the whole data set in memory. We just load in 1% of the data or 5% of the data at a time. And at each iteration, only the distances between the observations in the mini batch and the centroids are stored. And this makes it a very natural candidate for clustering with on-disk data. So there are some existing implementations. One is in R. This one, you want to be able to use these nice file formats. You can't, you have to have the data stored in memory. So that one doesn't work very well. There is one in scikit-learn called mini batch k-means. The data can be stored in an HDF5 file, which is awesome. However, there's like a step, very beginning of it, that just basically realizes the whole data set into memory and then proceeds with the mini batch estimation, which is not ideal. So we implemented our own version in R in the bioconductor software programming project. It's called MBK-means, and the idea is to simply replace k-means with a scalable version such that it can scale to really large data sets. So let me give you an example of its performance. We took a data set with 1.3 million mouse neurons, and we downsampled to smaller data set sizes ranging from 75,000 observations on the x-axis up to 1 million observations on the x-axis. And then we calculated the max memory used, followed on the left side in figure A, and the elapsed time in figure B on the right. There are 3 different algorithms that we compared, just regular k-means with Lloyd's algorithm, mini batch k-means with data read into memory in green, that's there in the middle, and then mini batch k-means in blue with data stored on disk. So what do you see? K-means is incredibly memory intensive and slow for data sets larger than around 200,000. And mini batch k-means, even if you don't want to deal with an HDF5 file, you have your data stored in memory, you can get huge cost savings in terms of memory and speed if you simply replace k-means with mini batch k-means. And if you want to store your data on disk, or you have your data stored on disk, you can cluster 1 million cells in less than 2 gigabytes of memory in under 10 minutes, which is phenomenal. And this blue line, for example, in figure A, will just continue to stay flat because the data are being capped out based on the user-defined mini batch size. Accuracy. So you might think there's got to be a catch. What's the catch? So accuracy is often the thing that somebody thinks about whenever you are wanting to make an algorithm faster. The take-home message is mini batch k-means is just as accurate as k-means for a large enough batch size. So we took a data set of sizes 5,000 starting on the left, 10,000 in the middle, and 25,000 on the right. On the x-axis, we have absolute batch size now. So I'm loading in maybe 75 observations, 100 observations, 500 observations into memory at a time. And then we're showing you on the y-axis the within cluster sum of squares criterion. And what you can see is k-means is flat, as it should be. There's no batch size for k-means. But mini batch k-means in blue and green, what happens is you just get noisier estimates for really small batch sizes. So if you only load in 75 observations at a time for a data set of 5,000, it's not as accurate as k-mean. But if you load in a batch size of around 500 or more, you get basically the same performance as k-means for a large enough batch size. And this continues to stay flat. So if we picked out a data set of larger than 25,000, that convergence happens from faster. We applied our mini batch k-means to the full 1.3 million mouse brain cells. I'm showing you a UMAP representation, which is like a low-dimensional, nonlinear dimensionality reduction method. I'm showing you a hex bin plot. It's a really cool way to visualize large amounts of data in 2D space. It basically grids up the entire plot into little hex bins, and then you can choose to plot the color of each hex bin based on maybe the majority of this like proportion of labels in each hex bin. So the colors here represent the different predicted labels from mini batch k-means. How did we do that? So we took our 1.3 million cells and performed PCA. We calculated the top 50 principal components, and then we clustered 16 different times to test out different K. That whole process of clustering took 3 minutes. The state of the art in R and bioconductor, either using maybe ScanP or the bioconductor framework or Seurat, is shared nearest neighbors followed by Leuven clustering for the R framework, and then BBKN followed by Leiden clustering. Those two processes took 35 minutes and 48 minutes total, respectively. And I will say that we also compared the labels themselves between mini-batch k-means and graph-based clustering and found very similar labels, and that is in the supplement if you want to know more about that. This is where you can find the paper in PLOS Computational Biology. Okay, so the next challenge that I'm going to talk about is thinking about how to scale up education for how to analyze single-cell RNA-seq data. So one of the questions that I often got whenever I started working in this area is how can I learn how to analyze single-cell RNA-seq data in a scalable way? So I wrote a paper called Orchestrating Single-Cell Analysis with Bioconductor with a lot of great collaborators, where we tried to describe many different things. One, standardized ways to store your single-cell data. We talked about workflows, about how to analyze them. And the thing I'm most proud of is this idea of an online book that was written in R Markdown. The book contains many different chapters, I think 42 chapters and 18 complete end-to-end workflows for different types of single-cell data. It has lots of philosophical discussions about single-cell analyses, and one I love is, what are the true number of clusters in my data? The answer is there is none. I love having that conversation with collaborators. There's lots of code that you can just copy and paste to be able to change out the example data for your data to be able to analyze your data. Okay, and then the last challenge I'm going to talk about is moving from single cell into this new area of genomics called spatially resolved transcriptomics. So I'll just state at the beginning that these technologies that you might have heard about, such as MERFISH, 10x Genomics Visium, SlideSeq, these technologies are really hitting their stride right now. In 2020, Nature Methods labeled spatially resolved transcriptomics as the method of the year. I believe that these technologies are here to stay, and there's a lot of great room. It's a wide open landscape for computational development to be able to help analyze and make sense of these data. So what is it? Imagine at a high level, we have a 3D representation of a human brain in Legos. As we go back through time and the types of technologies that we've been able to capture gene expression data, we start with bulk RNA-seq, microarrays, I kind of looped in there. Then I've talked about single-cell RNA-seq. Spatially resolved transcriptomics is the idea of being able to capture gene expression now in a 2D landscape. We are already well on our way going towards 3D, but at the moment, spatially resolved transcriptomics refers to this idea of capturing gene expression in a 2D landscape. And so with that comes a lot of open questions about how to analyze them. So I thought I would briefly introduce one example of a spatially resolved transcriptomics platform. This is called the 10x Genomics Visium platform. Shown in blue there, starting on the left, is what's called a slide. And then within the slide, there are 4 capture areas where you place the little tiny tissues, which are 5 millimeters by 5 millimeters in size. And so within each capture area, you have in red a fiducial frame, which helps you align an image with gene expression data together. And then within the fiducial frame, you have little tiny spots. There are 5,000 of them on this particular assay. The spots have these very special barcodes called spatial barcodes that allow us to have the RNA bind to them. So for every RNA that we captured, we have an XY coordinate where this bar or where this RNA came from in 2D space. So I'll point out that this particular assay has 5,000 observations or spots. Starting at the end of this year or early in 2023, we're expecting assays with 1 million spots in this same 2D plane. The idea is we're now going to be able to go from a spot that may capture anywhere between 1 and 10 cells to subcellular resolution. So we are quickly scaling up the data set. This is one sample, for example, and we're with my collaborators and at Hopkins at Broadly, we're generating tens of samples already. So that you can imagine like doing the math with like 1 million observations with like tens of samples. We're going to be quickly dealing with very large data sets. So here's an example. Okay. From a postmortem human brain, specifically the dorsolateral prefrontal cortex. So this is a well-studied area of the human brain. I'm showing you, starting on the left, this is a tissue that had manual annotation. So for each spot, a scientist manually labeled whether it was white matter or layers 1 through 6. See, and they're described by the different colors. And you can see that there's this purple color. Does this work? There's this purple color here. I'm showing you now the molecular representation from the gene expression space. So you can see that this layer is often represented well by this gene called PCP4. And you see two things. So you see the gene expression space shown as counts with the colors. And then behind it, you have an H&E stain. You have a histology image. So this is a really unique aspect to spatial transcriptomics data. The typical images that we see right now are either the histology images, such as an H&E stain, or an immunofluorescence readout. So there are lots of TIFF files that are being created with different channels, capturing different proteins, for example. And so I'm interested in blending two fields. One, thinking about spatial statistics and its applications to geospatial statistics, where they've really developed very robust formats to be able to store and work with these data. So GeoTIFF files. When you use Google Maps and you download or you load in, say, Baltimore, there's only like a small amount of data that's loaded to your phone, but as soon as you start to scroll, more data is downloaded. Those files are very robust, and I want to use them here to be able to quickly share images with collaborators, for example, and then borrow rich statistical methods to be able to apply to spatial, to these spatial transcriptomics data. And then there's this entire world of high dimensional and high throughput single cell data science, for example, mini batch k-means. And I want to combine these together to develop scalable spatial genomic methods. So just one last slide. Here's an example of a method that we have developed that was led by my postdoctoral scientist in my group, Lucas Weber. So starting on the left is a set of those manual annotations from that same tissue that I showed you. And then in the middle is a set of 6 genes. And these genes, if you look closely, you can see that they vary in space in a kind of a non-random way, right? You can see that MOBP corresponds to white matter, this dark region. This PCP4 I just showed you corresponds to this purple. SNAP25 is a well-known marker that maps to gray matters, layers 1 through 6. And then these 3 down here are well-known immune and blood genes. So I'm interested in finding fast computational methods that can identify what I'm calling spatially variable genes, so that biologists can then use them downstream in a variety of ways. So the approach we took is something called nearest neighbors Gaussian processes. There are a couple of approaches that you can use when you talk about Gaussian processes to make them fast. One, you can try low rank approximations. The problem with that is it approximates the spatial coordinates themselves. It uses a smaller number of spatial locations, which is not really what you want. Nearest neighbor Gaussian processes actually approximate the full data likelihood with just a smaller number of nearest neighbors. It's a really cool project, or a really cool idea that was developed by one of my colleagues in my department, Abidata. And so the benefit here is we can identify spatially variable genes that are scalable to large scale spatial genomic atlases, and they scale linearly in time. So all the existing methods scale cubic in time as a function of the number of spots. And so we went from cubic in time to linear in time, which is really great. There's an existing implementation in the bioconductor community called NNSVG. And that's it. I want to thank you for your time, and I'm happy to answer any questions.
We have time for questions. Yes.
Hi, my name is Jaime.
Let's use the
Yeah. Thank you.
Yeah, I'm kind of running by Statistics research on this area. So I read a lot of software here, and one of the big challenges is communicating what we've done and making our tools accessible. So when you're about halfway through your talk, you talked about writing a book for some of the work you did on cell clustering. Could you just elaborate a little bit more about sort of the motivation and the impact that that's had? In particular, I was interested in the philosophical discussion part about it. There's not a lot of room for discussing sort of like state of the art and contextualization like that. Thank you.
Thank you, Maggie. So great question. Yeah, so the motivation was essentially what you described. So every time a collaborator or a student would come to me, they would ask me these questions and I found myself repeating them over and over again. And so I wrote it down in a book. It just turned out to be 42 chapters at the end of it, which is really long. There's an entire chapter about clustering, as you can see here. There's an entire chapter about detecting marker genes and all the things that you would want to do in single cell. But the really cool part are the examples. So they're like end-to-end workflows with different types of single cell data, like full-length transcripts, barcode-based, site-seq, like you name it, all the different kinds of single cell data that you might encounter. And so it makes it very easy for a student or collaborator to come to this and be able to understand how to do it. But then the cool part is we explain why, and we compare different approaches in the book. Like if you try k-means versus graph-based clustering versus other like hierarchical clustering, like what are the benefits of doing one versus another? I find this to be like one of the most essential parts, communication of the science itself to anybody who wants to get into this field. Otherwise, it's just very dense. It's very hard to get into. Thank you for your question.
Yeah, maybe can I ask a question?
Yes.
Yeah.
So I think basically the trick in this hierarchy of clustering is probably in the tail behavior of the frequency distribution. So if you create, so because if it's a power law, it's probably a different behavior. You get a different behavior if it's, for example, exponential.
Alex, can you repeat the
Yeah.
The question?
So I think it would be really interesting to understand what governs whether the clustering works. And I suspect that it has to do with the tail behavior of the frequency distribution of the clusters, whether it's exponential or power law. Because when you subsample, if you have, for example, a small number of very dense clusters, essentially, you start to converge towards a Poisson distribution when you have a heavy subsampling.
Okay, I think I understand your question, but you can please correct me if I didn't. You're interested in understanding what happens with different type, like rarity of cells?
Yeah. So what is the tail behavior of the rarer cells, whether it's an exponential fall-off or is it a power law fall-off?
Yeah.
And you may need a different subsampling strategy. So.
Right. So I'll just answer really quickly. That essentially boils down, like if you have a data set that has really rare cell types, I would encourage you to pick a larger batch size. Even if you pick 50%, that's still a significant savings as opposed to 100% of the data in memory at a given time. But then you have the potential of capturing those rare cell types to be able to help you estimate the centroids. So if you're interested in broad cell types, you can get away with a small batch size. If you're interested in rare cell types, I would just maybe pick a larger batch size. This is a user-defined parameter similar to K in the choice. I appreciate your question, and we did think about this a little bit in the paper, and I'm happy to point you to it if you're interested in talking more.
Thank you very much.
Last question, Tom.
Yeah, I was just going to ask a philosophical question. I'm sorry. You can imagine this heading towards clinical science as well. For example, you can imagine someone taking out part of the tumor for the surgeon about where geographically cut. Do you want to comment on the timeframe for future development, like these things?
Yeah, that's a great question. So we're working with collaborators right now to apply this in ovarian tumors. And so ovarian tumor surgeries, for example, can take like 10 hours. And then you have to have somebody at 5 p.m. be willing to process that tumor for like the next couple of hours. So we have found this to be a huge problem actually in terms of wanting to take a fresh tumor and immediately be able to process it with spatial data. So at the moment, it's like a big problem, but I could imagine a world in which like we got that process down to like maybe an hour, in which then maybe a tumor could be used, or like maybe these technologies could be used in that way to help us identify where the tumor is. And if we caught it at the margins, for example, similar to the way you do like a biopsy for skin, for example. Yeah, good question.
One last question. Please use the mic.
something circumstances. Thanks for a very interesting talk. My question is, are there circumstances where the traditional KEYNOTE algorithm beats the new algorithm, or should everybody just use your new match KEYNOTE algorithm?
Yeah, I mean, it's hard to be able to make a statement that always, it beats it always. It's like a very a strong statement. I will say in all the examples that we have tried, which are a lot, it works really, really well. I'm actually using mini-batch k-means in application of spatial transcriptomics because, as you can imagine, we're now interested in identifying spatial domains in 2D space. And a lot of the methods are just very slow, like they take a week to run essentially. And so mini-batch k-means can give us an answer in 10 minutes, and it allows us to be able to move forward with science while we wait for maybe another algorithm to be able to run. And so in all of the applications that I've tried, it works really well. I would love to hear feedback when it doesn't work. That would be very motivating to me.
Outstanding. Well, thank you so much.
Thank you.
Very well. So next on the agenda, we have Peter Lawson from the Sheridan Libraries. Peter is a librarian for data and visualization in the Milton Eisenhower Libraries Services Department. He has a PhD in bioinnovation. And as mentioned before, Peter has joined our executive committee, and he's agreed kindly to provide us with an update on data services at the library. Peter.
Well,
Many thanks.
Okay. So thank you for that introduction. So hi, I'm Pete Lawson. I am in data services. We are a unit embedded in Sheridan Library. And we are, I think it's 1 moment. I was playing the slide timing and decided to go off the rails on me.
might be stay pretty close to mine.
So we're a unit in the Sheridan Library, and in particular, we're actually under the Digital Infrastructure Applications and Services Directorate in the library. So we're kind of the digital arm of the library. And we are the data services unit. So we're sort of the patron-facing unit in that directorate. So I just wanted to introduce our group as sort of a resource to the IDEAS community. So we serve faculty, researchers, students, even staff, and assist along the entire research data life cycle. So we help you find and access data, whether that's openly available data, whether that's JHU licensed data. We help you use data, so whether that's computational tools like in R and Python. We teach a number of workshops, or help you do geospatial data analysis in ArcGIS, ArcGIS Pro. We also help with data management, whether that's writing a data management plan for a grant, whether that's figuring out how to structure a research project. We can assist with that. Visualization. So I'm the data visualization librarian. So I teach workshops in data visualization in R and Python, as well as introductory workshops and basic design principles. And we also help with sharing data, whether that's a domain specific repository, whether that's the JHU data archive. We can help you take your data and your code and make it public, make it accessible, and give it a digital object identifier so that you can satisfy publisher and funder requirements. So we're a fairly diverse team. Most of us come from a research background, and primarily our main focus is around data management, as well as sort of research computing. And so those are the kind of the two arms. We also have a strong geospatial data analysis component. And somebody that's not mentioned here is also our consultant, Bryce Corrigan, over at Agora Institute, who helps as well with geospatial data analysis. And he also consults on spatial statistics. So if you're interested in spatial statistics, that's something else we can help with. So I just want to briefly go over sort of the service areas to show you kind of how we can help you. and then sort of the modes of interfacing with us, and then take any questions. So the data access and discovery, that's I'm also the data librarian. So if you're looking for data, whether you kind of know what you're looking for, but you haven't found it yet, you can reach out to us. We can help you find it. We can, provide strategies for finding openly available data, or we might know if it's been licensed to JHU, and we can point you in the right direction and give you instructions on how to access it. And then we can also do one-on-one consults to help you find data if we don't have access to it. So we've got a number of mechanisms for that. So one is the JHU Libraries Data Grant. So if there's data you're interested in and we don't have it at the library, we don't have it institutionally licensed, it's not open, this is a program where you can apply, and we have a small fund allocation to allow you to purchase data for specific research projects. Also the restricted data room. So if you need to have access to some data that is highly protected, for example, patient identifying data, and we have a space, the restricted data room, that allows you to access that data in a card or a JED card controlled room with an air gap to non-networked workstations. So you can satisfy all of those security requirements. So GIS and maps is one of our big areas. We can help you find and use geospatial data, use GIS software. We also have physical map collection, and we manage and administer the Esri site license. So if you're using ArcGIS Online, ArcGIS Pro, and you run into any issues with it, or there's anything you'd like to learn about it, we're that point of contact. And so in terms of data management and sharing, we help with data management plans. We help support compliance with funder requirements. This is going to be really big in 2023. If you are NIH funded, because you will be required to have a data management and sharing plan in your grants. We can help you prepare data for online access through the JHU Data Archive. This is our institutional data repository. So if your funder or your publisher requires you to share your data, share your code, this is one option. It's free up to 1 terabyte. And then we also specialize in human subject data. So if you are doing research and you need help with de-identification, figuring out what level to de-identify to, compliance with IRBs, we can also assist with that. And then, as I mentioned, we do a lot of computational research and programming. So we develop computational research skills through workshops, through one-on-one consultations, primarily around R and Python. We also promote open science and reproducible research. We run a reproducible research workshop series. So we teach Git, GitHub, versioning control, how to structure a reproducible project, how to integrate literate programming to your research. So using R Markdown or other reproducible document systems for typesetting literate programming output. Again, we're open source focused, so that's primarily Python, R, OpenRefine. We promote open tools, and this is continuously expanding. So this service area is just, depending on interest, depending on need, we continue to add new topics, new workshops, and new service models. So just a quick brief intro to kind of how you get access to this and what we can do. So the number one is really consultations. You can reach out to us at dataservices@jhu.edu. We do one-on-one meetings with researchers, students, staff, along whatever research data question you might have. But we can also do larger consultations. So if you want to meet with an entire department, entire research group or lab, learn a new topic or discuss a particular component of data management or setting up a reproducible research project, we can assist with that. And then we also do data management plan reviews, again, for that NIH directive, which is coming in 2023. So if you are doing NIH research, all data producing research funded by NIH is going to require a data management sharing plan. And we can help you write those. We can vet them for quality and completeness, and then we can help you in sort of the implementation too. So once you get that grant funded and you actually need to execute, we can help with sort of figuring out infrastructure, figuring out how you're going to do the sharing, and also how you're going to do, if you're doing patient studies, that de-identification and data sharing. And we also offer a ton of workshops. So I've got these links up here so you can go to these. And this is our link to our workshops. But we offer a number of open workshops. They're live webinars, so they're interactive. They're usually around 2 to 3 hours, depending. They're open to everybody that has a JED affiliation, so anybody that's a Hopkins affiliate. We also do workshops by request. So along these kind of themes, if you have a large group lab or something and you'd like to have a semi-personalized workshop, that's something else we can offer. We also have online self-paced training. So we're also recording all our workshops now this semester. So these will start going online live too, so you can follow along asynchronously as well. So we have partnerships with different groups for responsible conduct of research, and we run workshops through those as well, tuned to data identification, data management plans, things of that nature. So this is just a little subset of some of our workshops that we run. So a lot of it is introductory programming, data cleaning, data manipulation, data visualization. We also run research data management sharing workshops, like writing your own data management plan. We do a lot of GIS and mapping. And we do run, it kind of runs the gamut. So a lot of these are introductory, but we also run some more advanced ones. And we've run personalized workshops, for example, Hemi on creating effective scientific figures. So these can kind of reach across all domains here. We also launched a new series this fall called Data Bites. And these are short little hour-long lunch talks. They're sort of low pressure. You just show up virtually in Zoom and just hang out and learn something. And the theme changes every semester. So this semester is GIS maps and programming. There's a number of topics, but some of that, like speeding up your Python code, we try to seed it with things that are of interest to everyone. So it starts with really simple things like list comprehension, but it moves up to GPU computing. So it kind of runs the gamut. Then we also have, again, archiving and sharing, where we can help you deposit in the JHU data archive. So we can help you help facilitate that if you're looking to share and make your data reproducible and accessible. Help you select the appropriate repository. And then we refer you out if there's an appropriate collaborative resource that you could use, like Safe Desktop. If you have like patient data, we can connect you to the JHU's enterprise GitHub. If you aren't aware, we have an enterprise level GitHub. So you can create a GitHub account for your lab, for your department, and then also side server, obviously. So that's us. We're here to help you. Again, whole research data lifecycle. No question is a bad question. You can always just reach out to us at dataservices@jhu.edu. And you can grab these slides here if you want to see some of these resources.
Thanks, Pete.
Thanks.
On these slides.
In the research community, we to 3 types. There are usually last 90 days. And when we put that data into our longer storage, like 500 happens. And then we have a type of storage for the library of the. But there is a set of data, large amount of data that should be between 5 to 15 years because PIs is not keep of data. Is the library or can library do anything about keeping that data for so long?
Could you say that last bit, please?
So yeah, there is a quite a bit of data that has to be related with 5 to 15 years because people after 10 years or 15 years, I can go back to my data and do more research, applying technology, methodology, and extract more information out of there.
Right.
But there is a problem to keep that data for 15 years. So I don't know if I don't know offer this.
So that's one thing that you can use the JHU data archive for, because it is not archival in the sense that it's cold storage. It's just archival in the sense that it freezes the state of a project and has a DOI, a digital object identifier, uniquely mapped to it. So if you had all of the code, documentation, and data sort of wrapped up into that archive, then you can you could pull that down in 10 to 15 years, have a fully reproducible project that you can then start iterating on. And there is versioning on that data archive. So if you were to then publish new results and wanted to publish that updated version, you could. This does not link to SciServer. So this is an independent data repository. Is there a limit? Oh, sorry, limit on size. I'm sorry. Yes, so there's a terabyte limit for free deposits. And then there's pricing for the infrastructure beyond that, unfortunately.
Yeah, hi. So I have a couple questions in your workshops. So one is, what kind of attendance are you getting, and do you know where people are coming from? Because I've kind of wondered about how to reach out to, say, student body or something. And I see your workshops sometimes with emails. I don't know if you know where they're coming from there or the source. And also, if you take the opportunity to survey people as part of workshops and understand about data challenges and things like that, and if you do, are you able to share that information?
Right. So absolutely. So good question. So the first one, it's fairly so we receive about, I'd say in a good workshop, our more popular ones up to, I'd say 50 participants, and they usually range between, I'd say 20 to 40 generally. As far as participants, they're very diverse. We're getting a lot from KSAS, SACE, public health, school medicine. I'd say it's a pretty even split, school engineering, and I'd say, and even humanities. So I'd say it's a pretty even split, honestly, from most of the major schools. And we do survey them, and we kind of get a sense of, are we supporting their needs? What are their needs? How can we, build better support for them? That the sharing right now for privacy is internal, and we keep non-identifying information, so we don't record names or anything like that. So it is de-identified. So I think theoretically, if you reached out to us, we might be able to at least collaborate on maybe sharing out that information.
Sorry, I just want to come back to the question of data storage slide. So I run on London, campus, and we've got one lab who regularly generates a survival average revenues. And is it feasible for that kind of data to store your or we don't keep storing that going forward? And in total, we've got 2 and have 7.
Right. I think for the data archive as it stands now, probably not. I think we're expanding right now. We just released a new version that's going to have S3 backend. So we'll be able to start doing deposits into an S3 bucket. And I think that will scale it. But as it stands now, I don't think the infrastructure could support something that big.
The issue with S3 bucket, S3 bucket, you don't want this back. How expensive?
Well, what we're looking at doing is doing MinIO emulation over a server. Rather than going directly to S3, because that's the issue. It's like, it's cheap to put it in, it's really expensive to get back out.
Last question.
Just a related question. We do a lot with data and AWS, and are you prepared to help maybe with things that give us
So our support model as it stands now, we don't. So we're more on the kind of, I guess I'd say introductory to intermediate level computation. So for that large scale support, we're not touching AWS yet. It might be in our future service models, especially with the new AIX initiative, but not currently.
All right, well, let's thank Peter again. All right, next is one of our seed project awardees, Tom Lippincott. So, Tom, if you want to come forward. So Tom is appointed with the Alexander Grass Humanities Institute and serves as the Director of Digital Humanities. His work focuses on how machine learning, particularly deep neural architectures, can assist, inform, and guide scholarship in humanistic fields such as literature, history, and archaeology. He's been awarded a seed project from IDEIES, and we're happy he'll be able to provide us with an update.
Great. Thanks. about a year ago, I was at the Zoom version of this meeting, giving a description of some of the applied tasks that we were bringing distributional semantics to bear on, which is a machine learning approach that was developed in natural language processing and has been around for quite some time. And we wanted to bring it to a bunch of humanistic scholars who are working on various types of data. Today, postdoc Sheridan is going to be talking about how we're doing something similar, but for image modalities. So that initial distributional semantics study was very productive, and we had a lot of collaborations that came out of that. The same is true for now that we're opening up the world of computer vision and applying this to a variety of tasks that already exist and that people are already working on in the humanities departments in Krieger. And so we're going to talk a bit about, I'm going to tell you a bit about some of that applied research that we're looking at. We're at the stage of bringing to bear some of the recent advances in deep learning, some of the existing pre-trained models, and then fine-tuning them based on the particular needs of a given researcher. Yeah, I'll turn it over to Hallie to explain some of this.
Thank you. Okay, so I'll be talking a little bit about exploring computer vision models for their use in humanities. So we have at this stage collected a set of image corpora for 4 different domains as test cases, and I will go over them briefly and end with a few words on the next steps for this project. So the test cases, the first one is the handwritten correspondence of Maria Edgeworth, who is an Irish novelist who was writing at the turn of the 19th century on race, gender, and imperialism. This is a collection of her letters. The second test case, well, they all say one. Okay, the second one is in the domain of art history. So this is a twofold study. On the one hand, we're looking to identify Hebrew characters, and on the other hand, we're looking to see stylistic continuity and visual intertextuality in early Renaissance Italian paintings. And the third one is in the field of Near Eastern studies on the study of cuneiform, scholarship on cuneiform. And the fourth one is also Near Eastern studies on satellite images of terrain with dense archaeological remains in Yemen. So this is an example of a letter by Maria Edgeworth. So this is early 19th century. This photo was taken one of our team members at the British Library. It is very hard to deal with a large corpus of handwritten texts. So if we can find an OCR model that can do this accurately, that would really help the scholar and also improve the accessibility of these resources. As you can see, the letter was utilized in like multiple different angles and ways. So this is a challenging case for us. In this slide, you see 2 Renaissance paintings as examples for Hebrew characters. And there are 2 crucifixion cases. And maybe you can see they're not actually like the script is not complete. This is a very common inscription that can be found in the depictions of the crucifix in the Renaissance. In the Renaissance paintings, and it sort of reads Jesus of Nazareth, King of the Jews. So this is only one example of this. It's usually found in Hebrew, Greek, and Latin. And the other art historical test case is focusing on canonical artists like Albrecht Dürer here, and whose works have led to a lot of copying, forgery, imitation. And on the right, you see a reworking of the same image by Raimondi. This is a first pass that we did with one of our machine learning model attempts. And you see, even though it is identified as Cupid and a tennis racket, the point is it is able to replicate the compositional structure, which will help the scholar to identify more imitations of the same work. Here you will see a cuneiform tablet transliteration. So the cuneiform is the writing system for ancient Mesopotamian languages. it consists of a Latin transliteration of the tablet inscriptions, and usually Latin is not enough to capture all the nuances, so the transliterating scholars use a lot of diacritical marks. This is just an example, and especially in the secondary literature found before the 1980s, this is very hard for an OCR model to read accurately, and this would, if we could get one to read it accurately, it would really help a lot of scholars with access to secondary literature. And the final one is a satellite image in southwestern Yemen. And you see these high circular tomb structures roughly from 3000 BC. And I include the close-up of the two middle tombs, and then you can see sort of like this material cultural history of the material that was dug up to build these structures in those dots leading to the tomb. So this could, as you might have guessed, could lead to detection of more tombs based on the material tracks that was leading up to it, if it could be done by a machine learning model. So at this point, next steps for us is to evaluate how the machine learning existing state-of-the-art machine learning models perform, and then give them back to the domain scholars for more scrutiny. Thank you. I'm happy to answer if you have any questions.
Hi, Rob.
Hi.
So when we start handwriting images have different characteristics, and your classifiers have to require under some permutations, maybe orientations. Did you prefer to do anything to address this difficulty?
Well, our test cases do include, to some degree, linguistic components and to some degree more image-based components, but I think I'm going to refer to Tom on this.
Sure. So I think the point of showing these extremely diverse, as you noted, examples of is the common form of the computer vision task, which is basically that we're going to have people bringing in images that possibly have some sort of spatial annotation already on them, or that they want to have added to them, and having a sort of feedback loop between the processes of applying some basic form of machine learning model, whether that is something that is doing, like you saw with the DER kind of pre-trained object detection type stuff, or if it's something that is like a like we're using Microsoft's pre-trained handwriting recognition model to try to get that. At that stage, when we are getting that annotation back, we can do two things. We can either simply put those machine learning generated annotations back in front of the original scholars, have them tweak them in various ways, and we can just have some sort of generic fine-tuning process go on that feeds that back into the original model. Or I mean, my background is in NLP in particular, so when I see the OCR stuff, I think, okay, what is the language model doing? And I think that, okay, there's some very specific ways that we can fine-tune that, and that we can deal with the linguistic signal that is not correct for the particular type of materials that we're looking at here. And then when you look at computer vision, you often look at things like adding image noise or translations to the images to try to either improve the training process or to try to get different takes on the same image and get different, so that you can get those different orientations. Those things are all in the mix, but I think that right now the point of this, what we're working on right now is basically getting things off the ground in terms of all of these studies at once, so that we get a common baseline and have the common infrastructure for dealing with having these images and having this this loop, this process for the scholars to be involved in. And once that's there, we'll start thinking about what do we have to do specifically for this particular type of image or this particular type of content.
Good questions? Okay, thank you very much.
Thanks again.
All right, so I think last before the break, I believe
Yeah, okay. So we have a MARCC update. Jaime Combariza, most of you know Jaime. He has also an appointment in the Department of Chemistry, and he's going to give us an update on MARCC. Thank you.
Where's one there? It's one. Yeah. Yeah.
There you go. All right. Let me start by saying that in my previous life, I used to do a lot of support for the digital humanities. So I'll be happy to talk to you guys after this. So yeah, this is an update about ARCH, the Advanced Research Computing at Hopkins, and the new cluster called Rockfish. And hopefully I will provide some options. Maybe Stephanie, some of this could be good for you. So as I mentioned several times, this is a partnership between NSF, Hopkins, the Department of Energy, and then the 4 main schools at Hopkins, Krieger, Whiting School of Medicine, and Public Health. So our system is growing. It's now today close to 40,000 cores. And to give you an indication, on Monday, we added 5,000 cores, about noon. And by 3 o'clock, the system was 100% in use. So you people are making my life easy. We have different types of computing, what we call regular compute nodes. We call large memory nodes. We have a 1.5 terabytes of memory per node. And we actually have one node with about 3.5 terabytes by using Optane cards from Intel. And we have a whole bunch of GPU nodes with 40 and 80 gigabytes of memory per GPU. So there is plenty of computing for you to do your work. We have 16 petabytes of storage. All of that is a parallel file system, GPFS. We do not do anything in the archival world, so that's the library, and that's what I want to talk to you about. How do I get rid of this thing? Anyway. So last June, our cluster was in the 400 level in the top 500. And with this 100 nodes that we added this week, we should move up. And I was at a meeting Tuesday and Wednesday in DC with people that are directors of research computing. One guy from Purdue University was talking about his dissertation and what is the impact of being in the top 500 to the institution and to the research. I haven't read that paper. It's very interesting. But basically, we can use this to attract faculty and researchers, and also to attract funding. That's very interesting, and I like to read about that. But we have a very powerful system. We are adding more and more capacity and capability. So anybody at Hopkins, any faculty member, should be able to get an allocation on our system. We are giving the power to the PIs to do that. So this portal called CallFront, another NSF project, is going to give you the capability of requesting an allocation and adding users to your allocations. So we invite people to do that. All the links are in there, and it's very easy to use. And there are actually some videos that you can use if you are having problems using that. Some of the metrics from last year. Last year, remember, we have the old blue crab. Even though it was old, it was about 50% in use. So people still use a lot that system. So we have over 350 faculty members using or research group using the facility. That is a lot of people using the facility. We provided over 200 million of compute time. This is very similar to any of the national supercomputer centers. Over 400 publications, with over 15 classes were taught using our resources. This semester, there are 5 classes going on that are using our facilities. and lots of applications. This is an idea about the utilization throughout the years. Look at the last year, because we had the two systems in production, blue crab and the new rockfish. It was very high, in particular for engineering. Right now, we're at the same pace. So we're gonna provide more than 200 million hours this year, only with one system that is growing, and we still have capacity to grow the system. More about the rockfish model, because actually it has been a very good success at Hopkins. Remember, we're using the condominium model. So we use a grant, an MRI grant, to build the base system. And then we are asked deans to provide condos for faculty. They did that. And then every faculty member, or most faculty members, have provided small condos, large condos, and we have a very, very good system. So we started with about 400 nodes. Then the deans provided almost 200. And the PIs, 50 or so research groups, have provided almost 300. Actually, that number is wrong. Almost 300 nodes. So the PIs have contributed almost the same as the original system. So this has been a success that we have 40,000 cores that you people can use. And I realized, talking to new faculty members in Whiting, that I have to explain the condo model because it was confusing or they didn't really know what that was. So we have our base system, the MRI, and the dean's condo. And if Charles, he has a condo, so he's going to have an additional allocation. He has one from the deans, maybe one from the MRI, and then he's going to have his own allocation equivalent to the condo that he is providing. We have 5 or 6 new faculty members. They came with condos, 20 or 30 nodes, so they had that allocation for their people to do their computing. So this is the way how we increase the capability and the capacity of the system. Everything is shared. We don't have anything that is dedicated to Professor Don Carlitos. It's for everyone to use. It's only the allocation that is allocated to him. An example of, a PI gets an allocation from the deans, from the MRI, provides a condo, in this case, a GPU condo, so they are going to have an additional allocation equivalent to the number of hours of that condo. And people that contribute a condo, they will have priority in case that they run out of time during the quarter. They can get an additional temporary allocation, so they don't have to stop computing. Nobody has to stop computing. You just have to request a little bit more time. I will check if your group is using or misusing the system before I increase the allocations. Workforce development. So we have given a whole bunch of tutorials on how to use the resource, how to create or request an allocation and so forth. Last couple of weeks ago, we had a tutorial on the A-Flow framework for material science to do machine learning and to do a lot of computing. Next week, we're going to have another one from NIST called Jarvis, also in material science. So we are also providing topics in different research topics, and besides what the utilization of the machine. And I have to tell you about a new resource that you can use. We just installed a couple of weeks ago the popular application called Open on Demand. With this application, you can connect to the facility using your browser. And we already have a whole bunch of applications, many of those, most of them graphical applications that you can use. If you want to use Abacus, the application, you can just click on that one. If you want a Jupyter Notebook, if you want RStudio, you can do that. And I'm going to give you a few examples. So this is using MATLAB. So you can display this particular Mexican hat using your browser. Or in molecular dynamics, this is a trajectory. Some of you might be able to see a benzene molecule in the middle and a whole bunch of water molecules around it. So we can also visualize and produce a movie of that application. All of that being that in your browser. You are computing on your compute node. So you don't connect to the login nodes and you're not going to affect anybody else. So many people are very excited about using this application. And I'm going to end up with this. This is my thinking. So we are providing a whole bunch of infrastructure for the AI and training workforce. So you're welcome to ask any questions during the break or later, and I'll be happy to answer them. Thank you.
Thank you very much.
We will have time for a few questions for kind of here in the open. If anybody has questions.
Hi, are there GPU? So there was all the protocols for the contributed, are the GPUs that are off this cluster? Are the GPUs that are off this cluster?
Yes. So we have, actually, that's a good question. I forgot to mention that. We have right now 26 GPU nodes. Every node has 4 GPUs, either with 40 gigabytes or 80 gigabytes. These are the NVIDIA A100 GPUs, very powerful to do molecular dynamics or to do deep learning, machine learning, so forth. So yes, there are quite a few of those.
And those are in the Rockfish cluster?
Yes, everything is in the Rockfish cluster.
I'm interested in open on demand and its distinction from site server. It seems that there's at least some overlap in terms of the kind of the way you interact with it, and also the way you run notebooks on Jupyter, hub server or map sections. How are they distinct?
I do agree that there should be quite a bit of overlap. Open on demand is an application that has been funded for many years by the National Science Foundation. It's produced by the Ohio Supercomputer Center, and probably 90% of the academic research computing center offer that capability. So I know it's quite a bit of overlapping with the site server, and I think it's actually good.
All right, any other questions? Otherwise, thanks kind of very much. Okay, so now we have a half-hour break till 11:30. All right, thank you. Hey.
It's not till the afternoon, but I'm gonna just get it done.
I'm putting them on the desktop.
Great. Doesn't like it. What's going on? Oh, that's weird. Can I stop sharing it for a second? Doesn't want to. I think it's in. There we go. Let's make sure it did. Perfect.
If you're speaking in the next session, could you please check in your slides with the front desk near the door? Thanks.
Yeah.
So you can see how many people are just dropping theirs on the desktop. Yeah, so you can find it later.
Nice.
Okay. Oh, that's interesting. Oh, sorry, I didn't know that was live. Okay, should be good. I'm pretty sure.
Yeah. All right. Thanks.
All right.
Notice here, but I need to borrow that.
Oh, yeah, sure. There you go. Oh, and then I have your
Two different places. Yeah.
You can post it
I'm not sure. I got like.
Okay, folks. Good you.
Okay, folks, welcome back. So we have a selection of talks now before lunch, and the first one is an IDIES Seed Award report. The Seed awardee was Rama Chellappa, who can't be here. But the presentation is going to be by Soumyajit Ray, and the title is Development of an Artificial Intelligence System for Phenotyping of Patients with Acute Stroke. You should find right there. Talk.
Hello, everyone. My name is Soumyajit Ray. I'll be presenting development of an AI system for phenotyping of patients with acute stroke. So I'll briefly go over these, the motivation for our project, our aims, our overall approach, the data collection till now, and also our preliminary analysis. So stroke is the leading cause of death and the second leading cause of death and disability worldwide. And in the United States, every 40 seconds, somebody has a stroke. And every 3.5 minutes, someone dies of stroke. So this is the results of one survey where 38% of all people surveyed were aware of the major symptoms of stroke. And they knew when to call 911 to get help. And the other fact is that patients who are given treatment within the first 3 hours since symptoms onset are more likely to have less disability as compared to those who do not receive treatment within the first 3 hours. So this is how stroke is diagnosed today. So we have a stroke scale. The text is not important, what's in the scale. So it's basically a series of 11 items on the scale, which an examiner will go through one by one in the emergency room. So, and they will manually rate against each of these items. So there's a column on the right where they put their scores against each of these items, and they range from 0 to 4. And the higher the score, the worse off the patient is. So there's significant issues with this. So there is inter-rater variability. So it's the scoring is a bit subjective. Depending on the examiner, the score can vary. And there's also intra-rater variability. So there have been studies where the same examiner has been shown a video of the stroke patient at different points in time, and they have rated that patient differently. So it's very subjective. And also early assessment is very critical, especially in case of ischemic stroke. So this list of questionnaires is run through in the emergency room, but we could potentially do better. We could do it within the ambulance. We could actually take it even before we could provide it to a layperson if we can make this available in a manner which is understandable. And this is a stroke outcome. So this is measured by what is called the modified Rankin scale. What this measures is 3 months from the time of stroke. What is the person's independence level? So can the person live independently, or because of their disabilities, they would require continuous help? And this is measured using this scale again, which is again a series of questionnaires which the person asks, and the final score is obtained between 0 and 6, where 6 is the worst off. So that brings us to the aims of our study. So what we are trying to do is build the final product. The vision is that we want to build a smartphone-based application which any layperson can use. So, so let's say you suspect somebody of having a stroke in your neighborhood, in your family, and you turn on this application, and it will guide you through a series of steps. So you just point the cell phone camera at the person, and it records a video, and you ask the person a series of questions which you're prompted on the cell phone, and that bases the response. The responses will be sent to a cloud where our AI application will run, and it will give you back an immediate score of what is the likelihood of that person is having a stroke right at that point in time, and also what is the level of the severity of the stroke. So that's a end goal that we would get to. And the hypothesis is that so we want to record only the facial features, and the hypothesis is that all the information about the stroke can be extracted from the facial features itself, or rather changes in the facial features. And the second aim over here, is that we want to, at the time of stroke, like within 24 hours from the onset of the symptoms, there is sufficient information, we believe, that can predict what will be the outcome 3 months down the line, the modified Rankin scale that you saw earlier. So if we can tell the patient upfront what is the likely outcome, that would inform the treatment and the rehabilitation strategies. So this is a high-level overview of the project. So the stroke manifests in various ways. There is sensory impairment, there is motor impairment, there is speech and language impairment. And all of these manifests itself in the middle column that you see, where you can see the facial the changes in the facial measurements. And I'll come to that. What do I mean by facial measurements? There are changes in the expressions, in the eye movements, there are changes in the voice and speech. And all of these feed into our final aims, which is the statistical and machine learning based analysis that we'll do to predict the severity of the stroke. So this goes one level deeper. So what on the left-hand side, topmost left-hand side, you can see what we're collecting. This is the data set that we're getting right now. This is 2D images and videos of the patient. So we are collecting this data as soon as the patient comes in, but after their first treatment has been administered. So we don't want to interfere with the treatment at this point in time in any way. And after the first treatment has been administered to the stroke patient, we collect images and videos. And these are analyzed using our deep learning frameworks to get all these set of features. All these features form an input to our final classifier or regressor, which will give us the final score of the severity of the patient. So this is our data collection procedure. So we approach these patients, as I said, after they have been administered the initial treatment for stroke. And so we ask the patient to sit up or as much as possible. And then we use a cell phone camera, like a regular iPhone, to collect data. And this is just to give you a sense of what we are asking the patient to do. It's very simple tasks like big smile, frown, raise eyebrows, shut your eyes tight, fill cheeks with air. Then we record the eye movements, which we feel are very expressive of the underlying condition. And then we move on to some pictures. So this is a very famous sketch, which is used in multiple disease conditions. It's been used in Alzheimer's also. So the patient is just used to describe this image. And so what we are, and again, I'll come to this later again. So what we're trying to get from the patient is his or her comprehension of the image. So language skills, cognitive skills. And also there are specific indicators in the voice itself, which we'll use deep learning to discriminate between stroke versus non-stroke. This is another example where we ask the patient to just name these objects, read out these sentences, and just read out these words. So this is pretty much it. So in total, it takes about 10 to 15 minutes max for the patient, if all goes well. Sometimes doesn't. So this is a data collection till now. We target to collect data from 100 stroke patients, from also 100 patients who are evaluated by the brain attack team. So the brain attack team is in Johns Hopkins, assembles together whenever there is a suspected stroke or any other similar condition which presents itself as a stroke. So we want to collect 100 stroke patients. We also want to collect 100 patients who were evaluated by the brain attack team, but were not finally diagnosed as stroke. So we want to make so our machine learning or AI algorithm should be able to distinguish between these two categories, apart from also collecting data from 50 healthy participants who, we will contrast the healthy participant data with the stroke data. So right now we are in the early stages of our data collection. We have collected data from 5 patients, 23 patients who were not eventually diagnosed as stroke, and 3 healthy participants. So if you want to sign up as a healthy participant, please let me know. So this is the facial, this is the first part in my analysis. So this is the facial metrics. So these are pretty standard metrics. These are usually used for determining symmetry in the face, and facial surgeons would use such kind of metrics, but we kind of repurpose them to see how these facial metrics change for stroke patients after they've had a stroke. And so I'm not going to go into too much detail. So if you see the red lines are essentially the measurements that we are getting. And this is all deep learning based. So let me just go a bit ahead. So these are under 4 and these 2. So there are total 10 such metrics that we are currently targeting. There could be many more, but these are the initial focus areas. And so what you see on the left-hand side, this is an image from a publicly available database. So we use a deep learning algorithm to plot these 68 landmarks on the face, the red dots that you see on the face, and basis the position, like the XY coordinates of each of these landmarks. We calculate the metrics that you just saw, the 10 metrics. And on the right-hand side, you see a plot where the x-axis is the frame number of this video. It's a short clip. So every frame we independently calculate the metric, and you essentially see a trend of that metric across that video. So we believe, our hypothesis is that this captures very rich information about the facial movements. And this entire time series of the metric will be an input to our deep learning network, which will be the final classifier for stroke or non-stroke. So this is one such metric. We'll do the same thing for all of those metrics that you saw here. So I'll skip over this. We use a separate architecture to stabilize these metrics. And instead of using metrics, because once we calculate the landmarks and go on to calculate these metrics, they kind of constrain the information that we are capturing. So instead of using the metrics, we actually use another pre-trained deep learning network to parse the face, where we capture every region of the face separately and use a convolution neural network to process each region separately. The idea being that we would also like to make this entire framework very explainable. In the end, we should be able to say like which regions are most informative of our approach. Facial action units, again, basically these kind of movements specifically, so like inner eyebrow raise or outer eyebrow raise. So we, again, get these through a deep learning network, and these become part of our entire feature list. I'm sorry, I'm going fast. It's running out of time. A similar approach, again, this facial action units forms a time series across the duration of the video, and that again becomes our input to our final classifier. We use another deep learning network to track the eyes. So traditionally, people have used eye trackers, but we are using a deep learning network itself, which can accurately, to a pretty good extent, is comparable to like a full-fledged hardware-based eye tracker. And finally, we have the speech recognition part. So this is where we calculate the MEL frequency spectrogram of the speech of the patient. And we use a convolution neural network on top of this, through which we obtain features which are combined with the features that we obtain from the video itself, from the images. So this is like converting the speech or the sound into an image. And we also, this is in future, we also plan to use MFCCs and X vectors to calculate, to get features from these, the speech patterns. And this will be another input to the deep learning network that will do the final classification. So, so at this point, we're just, we're in the early stages of our data collection, and we're just kind of getting all our features ready, from the images, from the speech, from the language. And finally, we will combine all of this. Once we have a critical mass of patients' data collected, we'll combine all of this into a single deep learning network, which will give us the final output in terms of the severity of the patient. Thank you.
We have time for one or two quick questions. Yes, the roaming mic.
So
So if I would have this app on my phone,
Right.
my data set, if I calibrate, if I give a baseline, if I go through these things, would it make sense if I had to set on my phone to do the pre-training as a baseline, which will all then be more accurate? So for people with extra risk per stroke or something.
Sure, yeah, that will be much more informative for us. And I mean, while that would be helpful in the general use case, I don't foresee people recording the data upfront without any like concern, right? So we want to build the system to be able to do it without any kind of comparing before and after. But yeah, that will definitely be helpful.
Yeah, it's a really interesting question on this. You're releasing this publicly to everyone can use, like say at home, to think that or Alice is having a stroke. How do you deal with like the legal liability of this? In theory, all these people are going to be using this if they think their family members having a stroke because of classic science, like their face being grouped on one side. And so then if your app comes back, oh, hey, you're not having a stroke, but they are, that seems kind of almost risky. How do you deal with that liability, potential liability?
Right. So so I'm not a legal expert, so but from what I understand, so our approach has been that in case, so there'll be a significant percentage of cases where we are not sure, and we'll be upfront to tell that the app or whatever that is, it will say that it's not sure. Only when it's very sure, it will say that there is a stroke or there isn't a stroke. So we are okay with even if it 50% of the cases at this point, it says that I'm not sure, you need to consult a physician. So we are biased towards that. We would rather not make a wrong diagnosis.
Yeah, I was thinking about the same, but the opposite reaction. You get someone when you have this sort of and that is not right. You want to create mistakes on that person.
Right.
Okay, we have one quick question at the back. Then we must move on.
I'm sort of a similar maybe we should comment on any expectations generalized visual features or speech patterns that unscripted used. Is that a tool that doesn't work for people like that? And how does something inform research?
Right. Yeah. So the entire point is to gather a very large data set of a very diverse set of people, so that we capture all of those individual differences in our data set, and the deep learning algorithm learns that those are not the differences that it should focus on when distinguishing between stroke versus non-stroke. But yeah, that's a valid concern.
Yeah. Is there going to be a bias in your training by collecting the features after an initial treatment?
There is. Because I personally want to collect the data from the patients, and we have seen a dramatic change within a space of few hours. For example, if you administer TPA, which is intravenously administered to remove a blockage in stroke. So just before administration, the person was unable to say, he knew what to say, but he was unable to say it. And like in an NIH stroke scale, the difference would be like 20-30 points. So yeah, there is. And once we get some initial preliminary good results from our study, we plan to do it in the emergency room. We plan to collect data in the emergency room when the patient has come in before any treatment has been administered. But that has its own challenges in terms of how you can set up the system, etc. So we'll get there. Thank you.
Thank you. Oops. So next we have our second keynote speech by Faisal Mahmood. The title is Data Efficient and Multimodal Computational Pathology. Dr. Mahmood is an associate professor of pathology at Harvard Medical School and in the Division of Computational Pathology at Brigham and Women's Hospital in Boston. He received his PhD in biomedical imaging from Okinawa Institute of Science and Technology in Japan, and he was a postdoc in the Department of Biomedical Engineering here at Hopkins. So welcome back to Baltimore.
Okay, It's very nice to be back. And as you mentioned, I was a postdoc here in Dr. Nick Durr's group in BME, and also worked very closely with Dr. Alex Burris. So it's always a pleasure to come back to Baltimore around this time of the year, because it's already getting a bit cold in Boston. So today I'll be talking about some work that my lab has been doing for computational pathology. And I'll start by giving a brief overview of what we do in general and what the field of computational pathology is really interested in. So we're interested in analyzing a lot of phenotypic data. We're interested in analyzing H&E, multiplex immunofluorescence. And we do a lot of quantitative spatial analysis with the hope of getting to everything in the red box over there. So a lot of early diagnosis, prognosis, predicting response to treatment. We like to integrate information from modalities that are sort of already quantitative, you could call. So basically, one way to explain this is by using this nice. What we're trying to do is that we're trying to do everything in the red box here using a whole slide image, which can be very look something like this. A typical whole slide image could be between 100,000 by 100,000 to 150,000 by 150,000 pixels. So what can we do with this? Could we get to replicating what humans do, such as cancer diagnosis, other kind of disease diagnosis, prognosis, prediction of response to treatment to a degree, and can we go beyond what a human is sort of capable of? Features. And about 8 years ago, when convolutional neural networks were common, there was a big push to annotating regions of interest within these large images and using those annotations to train these models. But an issue with this approach is that you would need a lot of annotations, particularly to target more interesting, larger scale problems, which we definitely wanted to do. So a solution is the utility of some form of weakly supervised learning, where you could either build a graph on this entire image. It's a very large image. You could potentially segment all the nuclei or use patches and connect them, nodes and edges, finding perhaps similar features across this large image, and then use some form of graph convolutional neural networks. Or you could use some form of multiple instance learning, where you could assume that this entire slide is a bag of smaller patches, and then have some form of multiple instance learning following that. So we've been using multiple instance learning quite a lot, and one approach that we developed was clustering constrained attention MIL, which uses a number of different bells and whistles from conventional machine learning and brings it and sort of ties it together for pathology. So we use things like attention-based pooling, multiple attention branches, the utility of pseudo labels as the model starts learning, as well as using pre-trained encoders, whether from just trained on ImageNet, and the earlier layers of such encoders would perhaps learn some more generic features, and we could use those, or we could use self-supervised encoders. So the method is fairly simple, where we have a whole slide image. We can segment the tissue regions and then patch them into smaller patch regions, and then once we have those patches, we can extract feature representations from those patches, and this can again be from in the original study. This was from using like a ResNet 54, the first 3 layers of ResNet 54, followed by an attention backbone, where you would perhaps learn the most important features, something like this, and then you can have some other things to make things more data efficient, like instance-based clustering, where you can cluster more common regions together and then pool everything together to build a slide level representation. In the original study, we tested this on a number of different disease models. Here's an example for breast cancer lymph node metastases, which is a commonly used benchmark in computational pathology. So we trained this on the infamous chameleon challenge and adapted it to internal data from the Brigham and Women's Hospital. And we studied how data efficient this was, both for using just the TCGA as well as on independent test cohorts. What happens when we use fewer and fewer cases to train these models? Here's an example from going from using 700 cases to down to just 100 cases. And we did quite a lot of analysis in terms of looking at what regions the model was using to make these classification determinations. Here's an example for AE1, AE3 staining corresponding attention heat maps that the model uses. And there's quite a lot of quantitative analysis on this in the study, as well as examples on additional disease models. So what this approach really did for us was it was essentially a toolbox to train data efficient whole slide image level classification models. So we applied this approach to a number of different examples, and this method CLAM has been extensively used since we made it public. One example that we used it for was for identifying origins for cancers of unknown primary. So I'm sure a lot of people here are familiar with this problem, but for those who are not, when cancer metastasizes, it often becomes unclear where it may have originated. And this is a huge problem because most treatments are based off of the primary tumor, and these patients undergo extensive diagnostic pipelines, pathology and radiology, or a combination of those, other kinds of tests to identify where the origin is. We wanted to see, is it possible for us to predict the origin using conventional histology image, or at least get an idea for where the tumor may be coming from early on during the diagnostic pipeline. So the initial study design looked something like this. We had about 22,000 cases, a combination of cases from the TCGA as well as from the Brigham and Women's Hospital, and we grouped them into 18 common origins, looking at just grouping common tumor types together. And then we tested it on about 6,500 internal cases, a lot of cases that were sent from multiple medical centers using our consult network, as well as looking at cancer of unknown primary cases that were again sent from around the country, and doing some clinical correlation to identify cases that had some form of a primary differential design. So we used a CLAM-based architecture, but in this case, it was a multitask problem. We're given a whole slide image. We're trying to predict whether the case is primary or metastatic, as well as predicting what the origin is. So a binary problem and an 18 class problem. And there's some performance analysis. There's a lot more analysis in the study, but the model did fairly well for both tests, both on internal and external validation. But this is perhaps a more interesting analysis where we're looking at just metastatic tumors, and then also analyzing increasingly difficult metastatic tumors, because for cancers of unknown primary, it's not possible to have very clean ground truth. So the way a lot of the molecular assays on unknown primaries have established utility around this is by using increasingly difficult metastatic cases. We use things like number of ancillary tests as a surrogate measure, whether the case was poorly differentiated and so forth, to identify which cases are more difficult. And then we also analyzed what regions in the whole slide image were used in making those classification determinations, and then quantified everything in the tissue microarchitecture, correlated it with high attention regions to sort of quantitatively be able to say that the model was predominantly using high attention tumor regions to make those classification determinations. There's some more analysis on cancer of unknown primary cases in the study, as well as analyzing how top 3 predictions from the model can be used to reduce the number of ancillary tests that might be needed. More recently, we've also begun to integrate this with genomic information. So using our molecular assay at the Dana-Farber Cancer Center, we've combined histology and genomic information to see if we can predict origins better and how this improves. And this is a study that's going to come out soon. Another application we applied the method to was for endomyocardial biopsy assessment. So when after heart transplants, patients often undergo repeated heart biopsies to determine whether they're rejecting the donor heart. And this problem can have a lot of intra and intra-observer variability. So we wanted to see if computational analysis could help here. And the study design was something like this, where we had quite a lot of cases from the Brigham and Women's Hospital and from MGH, adapted it to internal data as well as a lot of external data. The model architecture was very similar to what I showed earlier, but again, it's like a multitask problem, identifying whether a case has cellular rejection, antibody rejection, and or has Quilty lesions, which are benign mimickers of rejection, and how it does for internal, how it performs internally, as well as how it performs when it's adapted to data from external sources using different slide scanners, staining protocols, and so forth. Another study that we have, another general direction that we're very interested in is combining data from multiple sources or multimodal learning. And in this particular case, for this study, we did a pan-cancer analysis by combining histology and genomics and trying to see whether we can use interpretability as sort of a discovery mechanism to discover new prognostic features in both molecular and morphologic profiles. So the architecture, this architecture is very similar to the CLAM-based architecture I showed. This is just a self-normalizing network, and then the fusion is basically a Kronecker product followed by two fully connected layers. These are trained individually, and then everything's trained end to end. We have whole slide. So we're able to get to prognostic profiles very easily, but perhaps the more interesting aspect here is that we're able to go back and look at what regions were used in the whole slide image, as well as what was used in the molecular profile, and then further quantifying everything in the tissue microarchitecture to the best of our ability gives us some insight. So we did this for 14 different cancer types. We're able to separate 10 of them in a statistically significant manner. We're also able to investigate for which tumor types was it beneficial to have a multimodal setup, for which ones it's just enough to prognosticate using the whole slide image, or and where it would help having genomics involved and so forth. And we did quite a lot of analysis on all 14 of the cancer types that were studied. Here's an example for clear cell renal cell carcinomas, where we're essentially using the whole slide image, and we can look at what regions were important in each one of them, and then how well you can separate the patients based on histology, genomics, and using a multimodal setup, quantifying everything in the tissue microarchitecture and how that looks. Another interesting finding that I'll refer you to the article for this is that we found that for 10 out of the 14 tumor types that we studied, the model on its own used tumor infiltrating lymphocytes as like a prognostic marker for lower risk patients. There has been quite a lot of analysis in that particular direction, but perhaps like a slightly more interesting aspect was that the model was able to make those predictions on its own. So we just feed it the whole slide image and the entire genomic profile, and the model figures out those regions on its own, as in comparison to some conventional analysis where the regions are defined earlier and correlated with outcome. Another thing that we have been very interested in is full slide level image retrieval. So I think many people here would realize that how this can be could be a very incredibly important tool. So supervised training is not always possible and practically impossible for some of the rare conditions. So we wanted to see if it's possible for us to have a large repository of whole slides, which are increasingly being collected as more and more hospitals around the country move towards digitizing their archives, as well as scanning these slides for clinical purposes. Given a new slide, can we sort of comb through the archive and find similar cases? And we wanted to do this in a way that it would be, essentially easy to set up and would be practically usable. This article already, it says to appear. This is an old slide, but this article came out on the 10th earlier this month. So we start by using the whole slide. We build a mosaic of features, mosaic of patches, and this is done just by using very simple k-means clustering as a way of using similar patches from the image. We use the autoencoder or self-supervised learning to have some form of feature representation here. In the study, we used the VQEAE, a vector quantized variational autoencoder, which was trained on the TCGA, and we extracted features using both the VQEAE as well as a DenseNet encoder trained on just ImageNet. We build we essentially build a Van and Bose tree. From this Van and Bose trees can be memory heavy. However, we found that the amount of memory that was required for this would what we had in conventional consumer grade workstations was enough. And essentially during search, you extract those features, comb through the tree, and there's a ranking algorithm, uncertainty-based ranking algorithm that we use to find similar patches, and then ultimately similar slides. So given a slide like this, we're able to find similar slides in a large database, as well as identifying what patches in the whole slide image did it use to make those determinations. There's quite a lot of analysis on this, on both common and rare conditions, as well as analysis on how the speed sort of scales with the size of the database. The speed, in this case, because we use a VB tree, the speed essentially stays constant. And this could be a very useful tool where we're investigating how it can be used also for other things like treatment. Do patients with very similar morphology have similar treatment outcomes and so forth? And the other direction we're essentially going in is trying to build like a multimodal retrieval mechanism. So essentially what we discussed so far is that if you have whole slide images and if you use multiple instance learning, you can essentially train very efficient models, classification models to replicate a lot of the existing diagnostic paradigms that we have computationally. But this setup has some limitations. So this is a nice slide that one of my PhD students put together. So one of the biggest limitations here is that this multiple instance learning based setup or attention ML based setup is not very context aware. So each one of these patches is mutually exclusive. They don't essentially communicate with each other. There's no communication between individual instances. So it's the tumor cells here, lymphocytes here. They don't essentially communicate from the perspective of the model. And then also we have limited data. But perhaps the most limiting aspect of the model is that it's not very context aware, which perhaps is enough for a lot of applications that we want to target in terms of diagnosis, might not be enough for other interesting applications such as prognosis and so forth. So we wanted to see what could we do about this. Of course, we know that histology, different levels would capture different kinds of information. And we wanted to see if we can use the setups that are commonly used for language-based models. So can we pose the problem as being a large document? So is our whole slide images more like large document where the context is important in terms of what goes where? So we built this method, this approach that we call hierarchical image pyramid transformer, where we have whole slide images, and it's a hierarchical self-supervised setup where we have 3 levels of self-supervised training. At the level of tiny 16 by 16 patches, and then at the level of 256 by 256 region-based patches, and then 4096 by 4096 patches, going all the way up to whole slide level aggregation. So this obviously has a lot of benefits because you're getting features at different levels, as well as a whole slide level aggregation that could be used for a number of different downstream applications. We did some initial investigation in the study for basic tasks such as subtyping, how data efficient it would be, as well as looking at survival analysis. But these are very basic tasks, and since then we've essentially explored more in-depth information around analysis around what could we do with these features. Could we use very simple learning on top of them to train models that would differentiate patients and so forth. So with that, I'd like to thank everyone who's currently working in the lab, as well as all the funding that we received to do this. And we're always interested in recruiting more people. So if you're finishing your PhD, please get in touch.
We have time for some questions.
I have great talk as always. My question really is about that last section, and it's motivated by pathologists reviews material today. And basically, we look at these large images in multi-scale. We have different magnification approaches on the microscope. My question is in that last section, it's a very computational a heavy job to do that large transformer in a powerful manner.
Yeah.
What about just doing multi-scale? Does it at least equal the college passing? You just say, we're going to try 4x, 10x, and 4x equivalent, all and mile still right with the same pinhole because you're getting the context by the scale rather than by the together.
Yeah.
What do you think?
Yeah, that's a great question, right? So essentially, just to rephrase the question, if you go to a lower resolution, it gives you more context, but perhaps less detail, right? So we try to do a lot of analysis on multi-resolution networks, trying to have individual networks work at different resolutions, combine them. So our conclusion about this so far has been that in principle, this is against what one would think, but 20x resolution, a majority of diagnostic tasks, it works perfectly. You could get very high AUCs. So combining multiple resolutions did not give us a huge benefit. But for prognostic tasks, it does give us a benefit, right? So the benefit of having the transformer-based architecture is that we could have a generic self-supervised trained model, which we can then use to extract features at different levels. So for some tasks, it might be enough to have it at a certain level. For others, it might be beneficial to have the whole slide level aggregation.
There was a confusion matrix for genes, and last gene was predicted as first gene. The two genes are similar.
Sorry, I didn't understand.
Okay. There is a confusion matrix in the presentation. You are showing different genes, performance of different genes, or any other gene.
Okay. No, it's not for genes. it's not for genes. It's for origins, right? So the confusion matrix is for origins.
Okay. Is this performance for estimated?
This is for origins, predicting origins.
This correlates estimated loss as well.
Yeah. So there's true origin on one side and then predicted origin on the other side, right? So the fact that the diagonal is being highlighted just shows that it's the performance is relatively good. I think that the confusion matrix itself is not enough to say this. In the study we have on the side of the confusion matrix, we have like precision and recall bars that you can look at for individual performance, right?
Any further questions?
I have two questions. The first, did you have any, or do you know if there is any data collection? Is it something like advantages or slides from multiple places? Is there a consistency with how it is, whether that it can be by a certain way that didn't get to the model? And then with the hierarchical transformer, it can be memory intensive, not very large. So how do you kind of scale with multiple levels of transformer? Do you down sample or just the raw balancing?
Yeah. So for the HIPT model, we basically did not downsample, right? So we did everything at 20x, right? And the highest level of it is very, very memory intensive and does take a very, very long time to train. But we were able to train a model in under a month. What we're trying to do now is taking several months because we've drastically increased the number of slides that we're using to train these. Initial study, the CPR article early this year used 10,000 cases from the TCGA. And now we're scaling up to 200,000, and then with the goal of using up to 1 million cases, which is taking a very long time to train. So we're looking into the efficiency. And I know the machine learning community in general is working on efficiency for transformers. We're looking into some of those methods. Yeah. I'm sorry, what was your first question? Oh, right. So we use data from multiple sources. So I think using data from multiple sources in general, what we've observed is that improves the performance of the model, because that makes it more robust, especially if there's redundancy across the same class. However, it's always the case that we have a single predominant data source, and then other sources are limited. So upon evaluation, there can be bias in the models. And I think one example of this that I showed was a very clean one, was what we had for the endomyocardial biopsy assessment, where we see that there's a drop in performance as we go from one institution to the other, and the data sets were deliberately constructed to have different slide scanners and so forth. So yeah, there can be a bias. Yeah. Okay, thanks.
Thank you. Let's Frank, what's next to Mahmood this time? For student fellowships, and we now have a couple of presentations from student fellows, and the first is Ryan Chou.
All right, hello. All right, so we'll do a 5-minute speed run for this. So we were looking at reconstructing surgery. So what we can pull, what additional information we can pull from surgeon instrument motion. So just some brief. Oh yeah, I'm not talking into the mic. Just some brief context to start. So our data is from a procedure, septoplasty. This is a very common procedure used to correct deviations in the nasal septum. The nasal septum is the structure of bone and cartilage that separates your nasal cavity into your left and right. And as you can see, there typically like a lot of there are a lot of different variations that you can have in deviations. So the one pictured is kind of like an S shape. You can get C shapes, and there are a lot of different ones that I can't really describe with letters of the alphabet. So, but you get the idea. And of course, for each of these different variations, you'll need to take, or the surgeon will need to take different steps to correct them. To make things more exciting, this is a minimally invasive surgery. So the surgical site is not directly visible to observers. They make an incision for closed septoplasties, at least. They make an incision just within the entrance of the nostril, and then basically do everything from there. So in terms of reconstructing the surgery, what we're doing is we are recreating a 3D model of the septum, like the structure, and the relative path of the instrument tip. And this is just using the kinetics data from the membrane elevation phase. So this is basically the phase where you have a septum and you have like the membrane, the tissue on either side. And the surgeon is taking a caudal elevator and like doing like a kind of like a scraping motion to separate it so they can work on the septum. So we kind of have a reconstruction pipeline. Basically, you start with the tool and the head sensor locations relative to an external tracker. And the output, I guess, is the tooltip motion relative to the centroid of the reconstructed septum. So sounds fairly straightforward, but also not quite. And the reason why is because live surgery is very chaotic. You know, you have differences in the anatomy, you have differences in the way the operating room is set up, in the way like the surgeon and the surgical techs have preferences for different instrument and sensor setups. And of course, so there are a lot of adjustments that we make. Also in live surgeries, we have errors in the data gathering process that you can't really correct. The priority is always the welfare of the patient. So we don't stop surgeries to make adjustments to the sensors and everything. So while this was validated on a cadaver study, the live surgery is still a work in progress. I did want to pull a quick sample from one of the ways we have been adjusting some of the things we've seen in the previous slide. This one is specifically a mathematical model of the instrument. So that's the Cuttle elevator with the sensor mount on it. The orange, this specific model is for the orangish cylinder. It rotates, unfortunately. So often you'll see like during sterilization or during different phases that'll move and it'll kind of destroy like the data. But this helps us recover it. We get different transform values to do that. And so, yeah, and so what we kind of get is a nice before and after. In the before, you kind of see the instrument just like the instrument motion floating off in the void. But in the after, with the correction, you can see how the instrument more or less enters through the same point, same incision, and then you can see the movement on the septum. So just some cases were able to be recovered with this method. So more excitingly, I guess, what does this enable? For starters, we can validate these reconstructed septums. We're getting the 3D model of this anatomical structure just from the surgical instrument motion alone. This is pretty big. It's like going in the reverse direction. We can apply motion data relative to the septum in surgical assessment. And this is we can pull features of, like, for example, the angles, the curvatures, consistency, things like that from this data and use it to assess residents. And maybe the most important or the most exciting one is the discovery of surgical strategies for motion. So surgical strategies can be thought of as maybe the instincts or the habits that surgeons have and how they change over as they gain experience. And so these surgical strategies will be how surgeons take prior knowledge. Maybe that's from CT scans or from patient history or from other factors, and they have like a game plan in mind for when they approach the surgery. And then how does this game plan change when they encounter obstacles or lack of obstacles? And then in the long term, we can quantify the relative difficulty of the septoplasty. In surgical assessment, obviously, we want to make sure residents are able to score well on difficult surgeries and easy surgeries. And in the healthcare system, and this can help us estimate the expertise needed, the steps needed, and the costs needed for different anatomies. And of course, the whole goal of this, I guess, is, or where we're headed is, we're now able to pull information, more information from the motion of an instrument. This will tell us more about the nature of surgeons, the nature of surgery. And the hope is that this will improve patient outcomes, the training and evaluation process of surgeons. Yeah, thank you.
We have time for one quick question when the next speaker comes up to the podium, if there is a question. Okay, thank you. So the next student fellowship presentation is from Keyi, if I pronounce your name correctly. Yeah.
Okay, so hi everyone, I'm Keyi. I'm a third-year undergrad student, and I'm happy to present my summer research with Professor Kevin Shortman. The topic is laying the foundation of large-scale precision stellar inference in the field of exoplanets. So the specific parameter we were looking at is the metallicity, which is basically measuring how much metal is there inside a star. In exoplanets, this parameter is especially important because it is directly related to the ability for a star to host planets. In the current research, we usually measure this value through a spectroscopic analysis, which is basically get a spectrum of a star and measure the lines in some specific wavelength. However, this method, even though it works effectively, it could be difficult and expensive to get a spectrum of a star. So we instead want to come up with a new method to use a data science approach to get a metallicity. So we try to fit a multi-wavelength photometric value to the isochrones. The isochrones is basically a line in the HR diagram that shows the stars with the same age but different mass and metallicity. And therefore, by fitting data to this theoretical line, we can guess we can infer some parameters associated with the star, like the mass, temperature, metallicity, and the luminosity. So the target we look at is open cluster. The open cluster is a loosely bounded group of stars for like about a few hundreds to a few thousands that were formed from the same dust cloud. So because they were formed from the same dust cloud, they roughly have the same metallicity, same chemical composition, and same age. So they become an ideal target for us to check our results. Because if we infer the metallicity of the stars and show they are consistent inside one single open cluster, we can essentially say that our method works. So we first collected multi-wavelength photometric data for all the targets we want to study. Photometry is basically you put a filter on the telescope and then take a picture of the night sky and measure how bright each star is. And it's much easier for us to get those values through large-scale surveys. And here are all the wavelength passband we use for the photometric value, because different surveys look at the stars in different wavelengths. And we try to synthesize everything and put it into our catalog. And then we use the Bayesian inference approach, which is basically update our hypotheses with the measurements. So we need to first come up with a set of prior probability, which are some things we roughly know about the star. So using the measurements of the star, we can get their physical parameters, such as how far they are away from us, roughly how old they are, and roughly how much dust there is between us and the star. So we defined a set of prior and use all the photometric value we collected to do a Bayesian inference and then use a sampler to get the final posterior probability. And we can use the final posterior probability to infer the star parameters. So this method is kind of not efficient in time because it usually takes about 5 or 6 minutes to fit a star parameter for a single star. And we want to fit it in a log scale for maybe like 2,000 stars. It could be a long time. And with the help of the IDIES, we developed a multi-processing tool to execute the code on the rockfish cluster, and it largely improves our efficiency. So here's a result of one single cluster called M67, which is an old and decent open cluster, which have a solar metallicity around 0. So we plot the metallicity on the y-axis and the color index on the x-axis. So as you can see, the result is pretty consistent. They are all scattering along the 0. And also we noticed that our infer result is especially sensitive to the availability of the UV band. So we color-coded the data points with the available UV photometric data. And as you can see on the left part, there are some white points with no UV band available, and they are kind of noisy in the plot. We also compared our value with other spectroscopic analysis to kind of confirm our result. And we compared those with the most GLAW and Apogee, which are 3 different spectroscopic surveys. And as you can see in this plot, the result is pretty consistent. They all show that the cluster have a solar metallicity, though for some spectroscopic analysis, there could be some outliers. But essentially, we have shown that our method is consistent with the traditional spectroscopic analysis, and the uncertainty is still within a reasonable range. Yeah, thank you.
Any questions or comments for Keyi?
Everybody just wants to answer.
Thank you very much.
Thank you.
Well, I agree. Lunch smells fabulous, but we have one more thing to do before we can eat, and that is to do poster madness. So please, can all of the poster presenters who are going to show us their posters come up, and you each have 2 minutes to give a presentation. Okay, please come forward. I think there's 6 of you in total. So please introduce yourself, state the name of your poster. 2 minutes total, and then we can all move on.
I forgot my name of my poster.
Oh, okay. So, yeah, stand in front of my friend. Identify the poster in some way. Oops.
Hi, I'm Jose. The name of the poster, if I remember correctly, a supernova battle, 2D CNS versus 1D CNS. Light curves give knowledge about temporal changes of brightness of astronomical objects. When Rubin Observatory starts its observation, it will have plenty of sparse light curves. For some of objects, we will not have data for some photometric bands. Therefore, we address sparsity using 2D Gaussian process regression, which is better compared to 1D GPR. And we converted each light curves to time frequency images named wavelets, and we did classification using 2D CNNs. The objects were type 1A and type 2 supernovae. We optimized our classifier using CP decomposition methods. We observed that rank 25 gave us better results. We also performed experiments using light curves instead of wavelet images, and normalization, initial normalization gave us better results. We found 2D CNNs significantly perform better than 1D CNNs. If you have any question, I will be here for answering. Thank you for listening.
Thank you.
Hello, my name is Faisal Navarro, and I'll be talking about the work my collaborators and I have done on combining and deblurring multiple astronomical images. Images taken from ground-based telescopes, they suffer from varying blur, which is induced by distortions in the Earth's atmosphere. And this blur can affect the perceived size and shape of the stars in the resulting image, which is problematic for astronomers trying to gather data on these parameters. And so deconvolution, which is the process of removing this blur, is an ill-posed problem, and it's complicated by the large amounts of noise and artifacts in these very long exposures of the night sky. So to tackle this problem, we modeled our observations as a random sample with a single latent image, and we tried to find an approximate maximum a posteriori estimate for this latent image. So we have multiple observations of the same part of the sky, and we're trying to find the latent image that's clean and not blurry. And the way we do this is we use the fact that an untrained convolutional neural network can impose a sort of implicit prior or deep generative prior on this problem. And so we develop a custom neural network architecture to extract information from each of these observations and then combine them into a single image, which we can then compare with our original noisy data by convolving with a PSF or point spread function. We test our methods on 33 images taken from the Hyper Supreme Cam telescope, and we find that they're cleaner, less blurry, and have the expected statistical properties that we would expect.
Thank you. You have slides for this one. Okay.
Hello, my name is Xiaowei, and I'm come from the School of Engineering. So here I will show how does the engineering, how does the data science related to the renewable energy and also the turbulence. So my talk will talk about we are planning to create the Johns Hopkins wind farm data set. This can be used for the community access to the high fidelity simulations. The reason this one, we know large eddy simulations has already can help the wind farms to improve the performance and the wind farm and also the operation. So, but when we run the large eddy simulation, we have a high fidelity simulation, we generate a large data set. For example, this figure showing here, we have the velocity field, the temperature field, even the pressure field along the different time frame at every position, means X, Y, Z position. So we have a large data set. So how to share this data set around the community is a big challenge. So we are targeting this challenge. What we are doing is we want to create a framework that can share the high fidelity simulation, large simulation result with the community. So everyone can use our data set with ease. So we have several goals. The first one, we need to make sure the database is public accessible, and then we include contains various variables such as the flow field information and also the forces on the wind turbine, wind blade, and also the energy generation. Also we would create a 380 terabytes database for public use, and with the new integration method to enhance the real world impact. So our poster shows the initial results outside. On the poster, we can talk more details later. Thank you.
Hi, I'm Han Shun Yao from mechanical engineering. My research is about turbulence. So we can see here turbulence everywhere in our daily life. Clearly, we can see in the clouds and this chaotic flow motion behind the car or under the airplane. So turbulence everywhere. One thing we're interested in is what we call energy cascades of turbulence. So basically, it tells a process that energy transfer from the large scale of turbulence to the small scale turbulence, then disappear because of viscosity. And this is the process I'm interested in. Thanks for the rapid development of the computer science, we are now able to compute this very complicated flow motion using supercomputer. But the issue is, how do we store this data? Because this huge amount of data can take probably like hundreds or thousands terabytes. With the help of IDIES, we have developed a publicly accessible database to store this data and we can do further any kind of analysis within this data. This database is based on Sci server, which can provide a faster and stable access to the data. In my work, I have used isotropic turbulence data, also apply a very novel statistical equation to explore the energy cascades within the turbulence. And what we found is that within the turbulence, the energy can transfer from the large scale to the small scale, and also can transfer from the small scale to the large scale. And these two different transfer mechanisms are related to different physical properties. And if you're interested in what's the property, you can come to my poster. It's outside, so welcome. Yeah.
Okay, thank you very much. Sorry, poster presenters. We're now done for our session. Please enjoy lunch, and the poster's outside, and we will reconvene at 2 o'clock.
Yeah, well, sometimes slow. The changes of a lot of study.
It's, yeah.
Thank you again. Yeah, that's a start. Yeah. Yeah, you're gonna have to do small, especially when you're not actually to do the early. Come along. Okay. Yeah, it's supposed to, I mean, yeah, there's a professional for a job and specifically I know. I think it's that. I know.
It's this one, isn't it? Get it out. You're welcome. I'm sorry about one more orthodox approach to service. Thank you, Mr. Chair.
Yes.
It was no problem. It was just here. There was part of the house. It was because there was.
Yeah, I think you guys are really.
I mean, I'm here.
Yeah. There are the most information. Yeah. As long as you don't think the day was rather than the United States. I want to get to word the word again.
There.
That's all right.
Yeah. Chicken, beer, meatballs, meatball. Yeah, you know,
Yeah. Yeah. That's why I know.
Okay. He's still getting to me, I guess, is what the difficult is. Okay. About the results and I don't have this. Yeah, that's gonna be my fun. Yeah, probably getting a little hold on.
This one's not, we have to get right up to all this stuff in and try to. Instead of. Seems like it's.
Yeah. That is.
Yeah. Like you can, it's a little awkward. It goes like that. Yeah. Yeah. Oh, yeah. Yeah. Yeah, I think it's the patterns. Yeah. We saw this year. Maybe next year I will be on. Yeah.
Yeah. Yeah. Yeah. Oh, yeah. Yeah.
Yeah.
Yeah, well, I did that on third part.
Oh, no. That's
See it.
You're asking one of the ways to put the you can look how you do better. Thank you, Gerard. I know
You look for, I mean, I was. Why do you do work? You like other people's money.
Hi, Susan.
You have a better idea what you're, hi. Good to meet you. I'm just trying to get my talk onto here.
Okay. Is this a laptop that was here and you just put it?
No, this is she put it, I emailed it to her and so I'm like, oh, now where's the stick? It's been too long since I've used Microsoft.
So yeah, go down, yeah.
Is it this one?
Yeah.
Day one presentation, is it there? No. I don't see it.
There's only one day.
Yeah. It's not in here.
Can go ask her.
Yeah, it had Malali in the name, that much I know.
So what, so this is day one right now that you've opened? No, there is.
That's day one, so unless it's in one of these.
3. Oh, no.
Session 4?
Oh, no, this is from last year.
That's not useful.
Yeah.
Schedules? No, none of these are directory. Phillips UFDD, right?
You know, I think I remember this. Can you try double clicking on Phillips? Because I didn't put it in a folder.
Oh, it was
Yeah.
at the top. Microsoft. Okay. It's been a long time since I've used Windows.
I'm the same way. I so I forget that that's a thing.
Okay.
It doesn't.
So should I drag that onto
the top one? And only shows one.
Okay, I assume you want me to put it on the actual computer. Is there a place you're putting these or it's just the desktop? Does it matter? I'm just gonna drag it onto the desktop.
Yeah, that's that. I think that's what people have been doing.
Okay. And then see if it's there.
Hooray!
Yay! And it's, okay, awesome. Thank you.
There has to be deep with like projects. Yeah. Yeah, so looking. Yeah.
Yeah. Yeah. Yeah. Thanks.
Yes.
Two-minute warning. Want to clap? Right.
Oh, yeah, speaking of which, right after, I think. Right after your talks? I should, yeah, okay. All right. These are for Combariza. All right. It's time to get started. Should I give everyone another minute or two? Doesn't look like everyone's back. But I hope this is not everyone. I need the whoever judged to give me the results too, because I don't know. Oh, you don't know who that is? Well, I don't have the results, so. I saw yeah. Well, as long as they get it to me before. All right, it's exactly 2 o'clock. I'm not waiting any longer. Okay, let's get started with the afternoon session. I'm Ani Takar, I'm the Associate Director for Operations for IDEAS, but more importantly, I'm the Lord of the afternoon. 1 session, 2 hours, no breaks. So I'm going to run a tight, oops, did I just shut off my own mic? Says the Lord. I think the Bilbo of the afternoon session. Gotcha. Yeah, what was I saying? No, it's still not working. The light flashed. Yeah. All right. So first up is our own IDEAS own Eric Mischang. He's from the West Coast, but he got his PhD way down under in Sydney, Australia. And then he did a stint at Amazon in Japan in the search engine division. So we rescued him from the dark side, so to speak. And he's been at IDEAS since 2019, and he's a lead developer for SciServer, and also an expert on parallel data analysis engines like Spark and cloud systems. So today he's going to give an update on SciServer. Thank you. And I'm happy to be rescued. For the record, is this? Maybe you're touching something on top that turns it off. I don't know. I just wanted to adjust it. Okay. Is that working now? No, the light is on. Yeah.
Yeah. Okay. There we go. Yes. Okay. Thank you. So I'll just get started. So I'm here to give updates on SciServer. I'm not going to give you training on SciServer or even describe exactly what it does. I'll just assume you know, or if you don't, go to sciserver.org, get an account and start playing around with it and talk to me. Okay, so I'll just start off by pointing out some of the recent hires that are either working on SciServer, supporting SciServer, or working on something that's peripheral to SciServer, like data sets or something. I think quite a number of them are here, so find them in the audience the next break, but I guess there isn't one, and introduce yourself. So I'll jump right into projects, SciServer related projects we're working on. So one of the things we're doing is SciServer, as you know, is a service that's installed at Johns Hopkins University here in IDEAS, but we also have installations of SciServer supporting sites around the world. And here's a new one. This is probably going to be the biggest external install once we get bootstrapped. We've just started working on this after quite a long talks with Brookhaven. They have a big budget. They're going to buy all new hardware. We're advising them now on what to do with that. GPU heavy workloads, petabytes of data. So this is going to be really exciting for us to get SciServer over here, and also opportunities for improving SciServer along the way for this use case. So another installation of SciServer that we have coming up is Division of Informatics and Pathology. So this is Alex Veris. He's a proponent of SciServer, and we've been talking also a long time on this, and we're actually making moves in this space right now. So we're excited to do this as well. And This is not a real solid agreement at the moment, but we've been talking with Durham, the Institute for Computational Cosmology. We've given them access to the software. They've started test install for just evaluation. So this is another thing that, this might be coming up in the future. So I feel like IDEIES is a little bit the Oprah of SciServer for you, SciServer. Okay, so another project, this is running on SciServer at Hopkins. We have an MOU with Elena Fertig from the Convergence, from the Single Cell Consortium of the Convergence Institute. This builds on a previous one where we designed some compute infrastructure for them to do single cell analysis and annotations using this Cell by Gene Explorer by the Chan Zuckerberg Initiative. So this MOU, we're trying to make this more streamlined for them, more embedded in the environment. And this just shows a demo of how you can go from SciServer dashboard to working with these data sets. And they're hosting a bunch of stuff on there. So yeah, look for more things like this into the future. So another development, we have the high energy astrophysics data. So we're hosting now the full HESARC archive of NASA. So this is basically their FTP archive hosted within SciServer, accessible via the cloud. And we have custom compute images which help you leverage this from within the environment. So this is something to look out for if you're in high energy, and hopefully bring a lot of users in that space over to SciServer. Another project utilizing the Chou SciServer for hosting data is the Additive Materials Benchmark series. So this is something, one of a couple of collaborations we have with NIST at the moment. And SciServer at Chou is going to host a mirror of the data and give access via the normal SciServer methods there. There's also been an extensive data modeling effort as part of this project by Gerard and Jaime. And we're going to be hosting 3D visualization tools within the SciServer environment. There's going to be documentations, examples, all of the things you expect from the science platform that brings it to this data set. Here's another one. Gerard mentioned this earlier and gave more details. I'm not going to talk about the project itself, but rather to say that while the project itself doesn't have a dependency on SciServer, we do the back end workflow management, and there's a API that hosts the data at TAC to big Tableau dashboards and the like to the agencies who are interested in this data. We've also made a lot of use of SciServer, and I think we might agree that it's been a big asset in getting bootstrapped and working on this project, understanding the data we're working with, and being able to really become a cornerstone of this project. And also, SciServer will sit at a place where we can provide the ability for a deeper analysis of the data for any of the agencies. A lot of them might just want the after products, but some people might want to take a deeper look. And the data hosted in the SQL backend on SciServer will give them that ability. So here's another thing that we worked on a lot before the AAS this year is the Apogee Fire catalog. This is a SCSS VAC in DR17. VAC is value-added catalog. It's mock catalogs of the galaxy. And they are accessible within CAS jobs. I think Sue had a whole hell of a lot of fun doing the ETL in a Spark cluster we have within the SciServer environment. And as a byproduct of this, we have CAS jobs tables, but we also have Parquet tables that are accessible in the Spark environment, should you be interested in that. And the FITS files are in SAS as well, but we don't expect people to use that much. How am I doing? Perfect. Okay, so I'll jump over to from the projects to software and service. So we're just a brief tidbit of usage. We're constantly growing. We have on the left side, there's registrations and logins. So we basically have steady, looking like pretty linear growth. Because a lot of that is actually people who come maybe a time or two, or crypto miners who want to use our platform for free CPUs. Probably a better representation is who returns to the platform. And we do have a core group of returning users after we kicked off all those miners. And you could see the numbers over there, and we have, I think I'm somewhere way off the chart over there, and probably several other of you people in the audience are there as well. Okay, so maybe what's bringing them here are some of these updates. One thing that we did face a fair amount of in this past year was these minors that I just mentioned. We've added a few things to help us squash them, and then that basically makes the platform better for everybody else using it, because the minors don't take up all the CPU resources and stuff. So we've added a bit more in terms of our ability to quickly squash them and detect them. We've added science domains. This basically gives you ability to kind of filter out what you see on the platform, because data sets like cosmological simulations can come with a lot of data volumes and a lot of databases that the typical user might not need to see. We have, specifically for some projects we worked on, we had user managed root volumes. We have several big data tools that we are able to deploy in SciServer, including Spark, Dask, all these accessible from SciServer notebooks, give you more power. SciQuery, that's an entirely another talk sometime. I'll just mention that the people working on that, Manu, Gerard, Stephen, are making quite a lot of progress. And that's the CAS jobs replacement, if you didn't know. So a few future directions. One thing we're working on is a consolidation of our UI. We actually, if you're not aware, we have several different UI components, makes it a little bit hard for us to make updates across the board. It brings in a few maintenance issues. So we're working on both improving some of the layout, the features, and getting rid of these developer issues by consolidating that UI and bringing it with modern features. One of the things we can do with that new UI, and we want to tackle a bit, is data discoverability. So this is what I am referring to as documents close to the data. So right now it can be a little bit hard to figure out what's accessible to you on SciServer. We have documentation on the website that's mostly static, and we can basically just document what's available to the public. However, having documents within the SciServer system, we can leverage the built-in access control. We can utilize this for projects. So for instance, the single cell consortium project with the cell by Gene Explorer you saw, we can basically create link farms to those apps and bring a more embedded feel, even more so, into the project. And what you see on the demo here, Juliana put this together. She's doing a lot of the work on the new UI. It's basically showing you how can you get from a login to SciServer to understanding where the data of interest to you is and how to get started working with it. So examples are one way that you might be looking to do that. So look for that in the future. We want to extend more of this one-click simplicity. We have several places along the SciServer platform where we have instructions, several step instructions, but making this a little bit easier. You might think that some of this could be, thank you, could be useful in teaching, getting students bootstrapped into the right environment very quickly. This has lots of extension points. It might be developing Jupyter widget-based apps, which I show down in the bottom corner there. Gives you an easy injection point to give a UI, but when you're not a UI developer. And this might be useful for projects that we work on in the future. So there's no avoiding it at this point. I think cloud technology is coming like a freight train. I mean, it already has been. And we don't really want to avoid it. There's lots of reasons why things like object storage are really popular, even in your own data center, but also the public cloud. So we've been working on understanding how we can hook in SciServer to cloud technology. And we've used AWS credits for this. We've Azure money as well from this. We have object storage nodes coming online within our data center that we're going to have to connect to somehow. We've increasingly gotten questions from folks that are interested in installing a science platform in the cloud. They already committed to cloud data, partly because they might get free storage posted over there for a science project like Hubble, for instance. How do we help them out? How do we bring that together? It's just obvious that that's going to affect the sustainability of SciServer in the future. We're going to move to Kubernetes. I don't say this just because Kubernetes is really cool. This is also the way that we provide external site installs is via Kubernetes. So this is what I call, and it's an industry term, is dog fooding. We want to be able to say, you know, we do this same thing that we're telling you to do, and you can trust it because we've done it here and we have thousands of users. So this is an important direction for us. We have a few other things on the radar. I don't think I need to go in detail here, and I think I'm done. Thank you.
Did you mention what the GPUs for that company?
Yeah, we've had GPUs for some time. None of this stuff is really public because it's difficult to give these kinds of resources public, because of course crypto miners are going to love that even more than our CPUs. But yeah, we're also building that out, but that's well integrated already with the platform.
Any more questions?
Did you show something of
Huh?
You show some slide about Apache.
Yeah.
What about the dynamics? This is about parallelizing the data.
Yeah, it's a parallel compute engine that runs on Java. It's this Hadoop environment, if you're familiar with it. So it's just really nice tool, really easy to get access. You can do interactive computation on a large cluster with it. And it was really useful for this particular project, just because we could go back and forth, and we had to do this several times, you know, adding columns and fields to the data. So.
This is a philosophical question for you, Gerard, but are the regional or servers?
I don't know if I should take credit.
So obviously, since the acceptance sizer tools in the lab, and a lot of them are very useful and easy to stand up cloud-based solutions for analysis closer to data, and some of the other problems this tax are solving. Looking forward to the future, like, could you tell us a little bit about what you personally think the advantage of using Sizer for data analysis, data hosting, relates to some like other solutions?
Yeah, so one of the things we've run into when we've talked to folks that may potentially want to have a science platform on their field is Jupyter Hub is a really popular option. So I think the likes of Google Colab is, I'm not sure if that's something that's generally installed elsewhere. I know that Google hosts it, obviously, but Jupyter Hub is a very popular platform for that. One thing that SciServer brings that's not as closely embedded in Jupyter Hub is the collaboration framework. And so we think that's a big advantage of SciServer. So really, I think that there's value there. And if we can get closer to some of the simplicity of a platform like Colab, which I think it's really easy to use. If we leverage these one-click things and make that a bit more streamlined, then we're quite close. And I think that, I mean, part of these tools are about simplicity. It's not just about compute.
I believe you have one other thing to take care of.
Oh, yes. So I'm going to announce the poster winner. Okay, so it comes with an Amazon gift card. This has nothing to do with me. I mean, not my previous Amazon experience or anything, but it's here, $100. And the winner is Fausto Navarro. And I don't have the title of the poster here, unfortunately, but I think it was about image smoothing in the night sky. You can say the title of your poster, but
yeah. Thank you so much. The title was Deep Generative Priors for Deep Learning Night Sky, I believe.
Thank you. Excellent. Well, great job. Okay.
So next up, we have first of the two keynotes for this afternoon. So the IDIES headquarters in the physics and astronomy building, which is across the street in the base of the Science Institute, which is where the backup is. So last fall, we kind of started sensing some tension coming from across the street. And of course, we were kind of very sympathetic with them. They were at the launch for the Webb telescope near. It was a job I did not do, but they had to get it right the first time. And boy, did they get it right. So when we launched on Christmas Day last year, the Webb telescope, we are very lucky to have an every project scientist for the Jaime Webb Space Telescope. Dr. Susan Valley with us today. So she's an expert on time series data and exoplanets, which is one of the mission of Webb. And her job is also to ensure scientific productivity of the Webb telescope. Which is pretty picture, but it looks like they're doing really well. And she has a long history in archiving data as well, and particularly because the archive and space telescope. So we're indeed very lucky to have her, and she's been the title of her talk is the Webb telescope. The data behind the images and why the images we had so far.
It's here, I promise you. It's just down 30 layers. Whoops. Did you see it pop up?
Yeah, it was
That one.
go back to the.
Is it in here? So you can tell my ability to use Microsoft has gotten poor. There it is. Hopefully it'll run okay off of the USB stick. Let's pick up slideshow. No, this is fine. Thank you very much. Thank you all so much for inviting me to tell you about the James Webb Space Telescope. I now see that you maybe even have an older talk, so I might get a little flustered as my slides changed order, but it'll be okay. Yeah, I am the JWST Deputy Project Scientist at the Space Telescope Science Institute, and I am also kind of transitioning over to be in the Data Science Mission Office there as a mission scientist, where I'm going to still be working on James Webb, but also thinking more about other ways of dealing large data sets coming from NASA. So everybody at Space Telescopes up, they want something about JWST and data. Susan, that's you. And I'm like, all right. Let's start. I know we're a diverse audience, so I'm going to attempt to bring astronomy to everybody, but still talk maybe a little bit more in depth about the data. So if you don't know, the James Webb Space Telescope is a 6.5 meter telescope that we launched into space. And if you have been paying attention, on July 12th, we released our very first images to the public. And here's the first one that was released. This was actually presented by President Biden the day before we actually wanted to let all the data out. But when the president says, hey, I want to be the one to release the data, you say yes. So what you're looking at here is a long stare, not even all that long, a few hours stare at a small patch of sky. And you see actually that blurry bit in the middle, which is a foreground galaxy, which is actually lensing the light from distant galaxies. So they're coming up and the light is being bent gravitationally around, which is actually causing those arcs you see. So it's a lens, a gravitational lens. But every smudge you see on there, except for the basically 3 stars that have big, long diffraction spikes, are galaxies. So you're looking at billions of stars in each one of those smudges. What was so great about this image is it showed us that Webb was going to be able to complete its mission. And one of the primary reasons we launched Webb in the first place. And that was so that we could see galaxies that formed right at the beginning of the universe. And if I can remind you, if light has traveled, travels at a finite speed. So that means that if it was emitted from a galaxy that formed at the very beginning, it would take 13 billion years to get to us. So if we're looking at a galaxy that's 13 billion light years away, it took 13 billion years for that light to get to us, which means when we're looking at these galaxies, we're actually looking back in time and seeing the universe when it was in its infancy. Now, the most distant galaxy in here that record has already been broken several months later by teams who are trying to do this as their science. But this was a demonstration that Webb could succeed in its mission. So let me take you back and just tell you a little bit more kind of about how we got to this point and the data that makes up images like these. And then I'll put some more pretty pictures in to keep you all your attention. So Christmas morning last year is when we launched the Webb Space Telescope. This is the Ariane 5 rocket that Webb was put inside of. You maybe have already noticed, wait, you got a 6.5 meter telescope with tennis court size sun shields into that fairing at the top of that rocket? And I say yes, because we had to learn how to fold a telescope in order to send it into space. This is one of the few times I saw a silver lining in COVID, is that because of COVID, I couldn't go to the Institute to watch this launch. I was not asked to be there at all. As a result, I got to watch it with my kids at home, which was really nice and meant that we got to have Christmas. And what a great Christmas present this was. This is an animation of the last stage. So you can see we have this folded up telescope. And what was really nice is they put a camera on this last stage that as it pushed it off into orbit. And this is the image of Webb as it was sent off to L2, which is where it was going, where the solar panels are just being released, which happened right at the beginning. The first possible moment that they could deploy those solar panels, that's when it happened. And we knew we were getting power to our telescope. Then over the next month, the telescope unfolded. Step by step, a lot of nails were biting throughout this period. There's these huge sun shields that had to unfurl. The thing that holds the mirrors had to come up, and the wings of the mirror came forward. So the mirror is not one piece. It's made of 18 segments, each plated in gold to reflect infrared light. And it did this spectacularly. There was almost no problems at all, even little tiny ones. This just went very smoothly. And it travels out to a place called the second Lagrange point, which is 1.5 million kilometers from the Earth. What's great about this point is the place where we can kind of keep a nice stable orbit, but it stays in an orbit around the sun that stays in lockstep with us, the Earth, which means we can keep communicating with the telescope, which means we can get down more data. So, but what's also nice about it is it's far away and we can keep this telescope incredibly cold, which is very important to Webb's mission. So we take a quick look at our telescope. We have a primary mirror made up of 18 segments. Each of them is about 1.2, I believe, meters. So pretty big, each one of them, to make up that 6.5 meter diameter. We wanted a big mirror like that to collect lots and lots of light. But you see those big sun shields, we have 5 of them. They keep the mirror side, and then on the back of the mirror, you have all the detectors, all our cameras are on the back. They're all kept incredibly cold. That side of the telescope stays colder than 50 Kelvin. And there's parts that are down to around 7 Kelvin. One of the instruments is additionally cooled. And then on the other side, we have the hot side of the telescope, which is several hundred Kelvin. And you have, that's where we communicate with the Earth. So that's always pointing down towards the Earth. We don't want to turn the telescope around and point at the sun ever. No, you're always pointing out into space. It's also where we have the thrusters so we can control the telescope, and the solar panels to get the light to power the telescope. The reason we want to keep the whole thing extremely cold is because we're observing in the part of the electromagnetic spectrum called the infrared. Now, if you know anything about infrared light, you probably know that everything that is warm glows in the infrared. And so just like this lovely picture I found off the internet of this man glowing in the infrared. And so as you can imagine, we have to keep the telescope incredibly cold because we want to see galaxies on the other side of the universe, and we don't want to just observe the telescope itself, right? So we have to get the backgrounds down really low. And the way we do that is get it really cold, hence the need to go to L2. But the other thing that's really great about infrared light is that it has some different properties. For instance, you can see in here that the man's hands glow through the garbage bag, which is something you can't see in in optical light, like we can observe with the Hubble Space Telescope. And this has nice properties for observing things like baby newborn stars. This is where I wish I had my original the talk I had edited yesterday, because if you were paying attention yesterday, they put out the Carina Nebula, and so I inserted it here, but I only did it yesterday, and that's where this major talk changed. But instead, not the Carina Nebula, the Eagle Nebula, the Pillars of Creation, the Hand of God image, if you were seeing that yesterday. And this is another great example where they compared Hubble and Webb to each other, but you can see it here as well. What you're seeing here is like, this is a portion of a large nebula that's like 6,000 light years away in our own galaxy. And there's these gas and dust in here. And if you compare Hubble's image to Webb's, you can see places where there are stars you can now see that you couldn't see before. And so this allows us to see the very beginning of star formation in ways that we never could do with Hubble. There's also the advantage of that Webb has better resolution. And so you can start seeing things like the waves and the turbulence and the gas and the dust that you see in that nebula. Okay, so the data. The data starts with the instruments. There are 4 instruments aboard JWST. There is NIRSpec, NIRCam, and NIRIS, which all observe in the near infrared. So talking 1 to 5 microns. And then we have one mid-infrared detector called MIRI. So all of these pretty much do a wide variety of different ways of observing and slicing and dicing light. What you've been looking at is the imagery, but they also all do a lot of spectroscopy. So I just mean taking the light and putting a prism behind it and spreading it out into a rainbow so that you can detect how bright things are versus color. And with spectroscopy, we can do things like chemistry and physics and measure precisely what things are. So NIRCam is where our image is, where we started. That image I started with the big galaxy at the beginning. NIRCam is the one that took that. So it's our main imager. It was also the one that used to align the telescope. I will get back to our multi-object spectroscopy and some of our spectroscopy modes in a moment. That data is stored at the Webb, up on Webb on a hard drive. So after you take an image or a spectra, we store it up on Webb, up at L2, right, on this hard drive, which it's really only a 68 gigabyte hard drive. It's not that big, it's tiny. But you know, if you're gonna launch something in space, you need to know it's reliable and it needs to just meet requirements. NASA doesn't really think towards, this is going to take us 20 years to build. Let's have, look towards future things. So we've been building this for 20 years. So that's what Webb has to work with. We have a 68 gigabyte hard drive. We pull down about 60 gigabytes of science data then every day through the Deep Space Network, which pulls it down at about 28 megabits per second. That's like broadband speeds. That's not too bad. It then sends it to Baltimore, where we then, at the Space Telescope Science Institute, where we are the science and operations center. So people are up there with their headsets and they're sending information to control the telescope, but they're also getting all the data and giving it to us in Baltimore. And that's where we process the data and run the JWST pipeline, which it basically takes that volume of data and doubles it and sends it to the Mikulski archive, which is the same archive that holds all the HST data, as well as data like Kepler and TESS, if you know some of the other NASA missions. The Mikulski archive, known as MAST, it then works its best to change that data, makes that data really accessible to astronomers so they can go in and grab it and do whatever science they want. So some of those scientists are at STSCI or at JHU, but others are around the world. And
But so while I showed you the flow to the astronomers, there's actually a backwards flow of data as well that's worth mentioning. With the green arrows, astronomers also have to say where we want to point the telescope. They're the ones who put in the observing proposals and says, this is the great idea I have. And so they send that to Baltimore, who then writes up a whole bunch of scripts that says, okay, Webb, I want you to, point here and expose for this long. And that goes back up through DSN to tell Webb what to do. Astronomers also love to make high-level science products, like they modeled something, they got the data perfect, and that's how they made their measurements, and they send that data to MAST as well, which MAST then holds on to and makes available for everybody else. That happens as well. What was interesting about this loop is that during commissioning, the fact that we had to observe, get data into the archive, get it to astronomers who then would have to respond and tell the telescope what to do would have to happen within a 24-hour loop. Because at the beginning, if you remember, we have those 18 mirrors, and the 18 mirrors had to all be aligned to go from 18 stars on our field of view down to 1. And it had to come perfectly aligned. And this happens through an iterative process of, oh, that's where that thing is. That means I should tilt the mirror this much. Oh, it ended up there. Okay, let's do that. And so it was a loop that had to happen. So imagine a bunch of people, astronomers and engineers who normally have normal, very day jobs, had to be up all night to complete that loop and ensure that every bit of that process was always working. You couldn't just suddenly have a computer go down. You needed to have someone there to reboot it if it needed to happen. And so that's what was happening during commissioning, is that both the yellow and the green loops were going on constantly. Okay, so we got the telescope into focus, yay. And that allowed us to take a picture like this. I've already showed you Webb's first deep image. The reason we're looking in the infrared was because we wanted to see galaxies that were at the edge of the universe, like I already told you. The reason they're in the infrared is because the universe is expanding, right? And so as the universe expands, that means that as that light is traveling from this galaxy, it's actually, all of its light is redshifted from what was probably emitted as ultraviolet or optical light into infrared light. And so that's why we observed in the infrared. And so we have the NIRCam telescope. And so astronomers then take data like these. So I wanted to kind of step you through the data and how you get to an image like that so we can make those measurements. This is actually the full field of view for NIRCam. It's actually 2 detectors that we have. And so there's actually a cropping that is done on the image that you see. But one thing to keep in mind is we actually observe at individual filters. And each of those filters just measure, you send the light through a filter, you only get that light, and that light just shows up as brightness across the sky. And so that happens for, this image is made up of 6 of these filters, like at 0.9 microns, 1.5, and 2, and they all get combined to make that color image. Additionally, each of those images is actually made up of a whole bunch of other images that are averaged together. Because we're using these near-infrared detectors, they have gaps in them and detector flaws. And the best way to average that out is actually to move that image across your pixel plane. And so here's a cycling, we call this dithering. So you dither around and then you average them together. And so all that information is in the archive as well. Additionally, each one of those individual dithers is made up of measuring the data up the ramp. So you can actually, as the photons are landing in my detector, the well increases and you're measuring how bright it is versus time. And it's actually that slope that you measure as how bright it is. So how fast is it gaining brightness? And those ramps are also stored in our archive. And so that's the data processing is going from those ramps for each dithering, averaging them all together, and then combining them, into a beautiful picture. So there's many steps to the processing to do all that. All right. So as I told you, I'm going to tell you about spectroscopy as well, because this is one of the great innovations. And the truth is, Webb is actually observing almost 70% of its observations are spectroscopy and not imaging. So what goes out into the public is almost always images, because everybody really understands that. But spectroscopy is actually the workhorse of Webb. And one of the great innovations on Webb is it has this multi-shutter array on the NIRSpec instrument. Each of those little boxes represents a little shutter that a little magnetic arm goes over and opens and can open and close each of those shutters. This means that we can let the light through for just the stars we want across our field of view. So a little magnetic arm opens them just where like the little blue stars are, and then we put a disperser on the other side to create the rainbows. And that way you don't get rainbows on top of each other, and you can just get the spectrum for the stars or the galaxies you want. You then measure the brightness versus wavelength, and you get images like these. So they did that for galaxies in that original field. And so what you're looking at the top is the brightness versus wavelength out in near infrared light for 4 different galaxies. And you can see that they each have these emission features of hydrogen and oxygen. I know it's kind of hard to see in this image, but the same pattern occurs at different wavelengths. And that's because the light is being redshifted, like I said. So the farther away it is, the bigger the redshift is. And now we can measure how far away our galaxies are using spectroscopy from the NIRSpec MSA. Another thing that a lot of people are using spectra for is actually to measure time series spectroscopy. And this is primarily being used to measure the atmospheres of exoplanets. And this is another way to kind of slice and dice data. So you maybe have saw this. This was released on that same July 12th this summer. And it was said, we were able to detect carbon dioxide in a giant planet known as WASP 39b, orbiting a sun-like star, it's some tens of light years away. So this actually seems like an incredible feat to me, and the fact that I'm able to say that we detect carbon dioxide on a planet far, far away is amazing to me. And this is because this planet actually transits its star. The planet goes in front of the star and blocks the light. With Webb, we measure how it blocks that light as a function of wavelength. And so another way to look at that same data is here, and where you're actually looking at, here they've broken it down to just 3 different wavelengths, but we actually have it for each one of those white points you see on there. You actually get a transit. So this is planets going to front, no stars, planet's not blocking, planet blocks, that's the transit. And so it gets dimmer. You notice that it gets the dimmest for that green set of points. That's the 4.3 microns, which is where that carbon dioxide bump is. And so you're actually taking this hundreds of spectra. Perfect, thanks. So that's how we get to these transit spectroscopy results. What's going to be really cool for Webb is that this is for like a huge giant planet. Like this planet is the size of Jupiter, but it's actually the mass of like Saturn. So it's this big puffy thing that's in a 4-day orbit around its star. But we're gonna be able to do this with a little luck for even planets that are the size of the Earth that we have found. They're orbiting very small stars, which boosts the signal to noise, but we will actually start to be able to measure the atmospheres of Earth-sized planets. Okay. So that is our time series modes. There's loads of these time series modes, and so we'll see what comes out of those. This is all processed in our pipeline, and this is, I promise, probably maybe the most boring slide I have in here, but maybe not for this group. This group loves it. It was actually the difficult thing we wanted to be able to do is have a pipeline that ran that we could then provide the products for everybody. But then when we get it kind of wrong, we didn't set the things just exactly right, that they could run it themselves. So it's actually an extremely modular pipeline. So you can run just 2 to 3 if you want, and you can run it on your own laptop. It's all written in Python, and it all calls the same calibration files, etc. So that was one of the great innovations. what we're having challenges with right now is that, as you might imagine, real on-sky data is not exactly like the simulated data that you have. There's artifacts and scattered light and strange cosmic rays that show up. And as a result, as we keep learning about these data, we keep improving our pipeline and how we best calibrate those data. And so every time we learn that, every month or two, we improve the pipeline, improve our calibrations. We then have to reprocess all the data to provide all those data products to people again. And right now it's happening faster than we can reprocess the data. I'm expecting this to slow down, but luckily everybody can run the pipelines. We just hand them the new calibrations and then they can do it. I would be remiss to talk about web without talking about the data archive, since I am now the deputy PI for MAST, so it's one of my favorite parts. The Mikulski archive is the home for JWST. All the data goes in there. Know that all JWST data goes public in a year, and a lot of it is public immediately. All those images you're seeing, that data is entirely public. It's in FITS file formats. You have to like those, but other than that, you can get at it. And some of the data is even, you can explore it and without even downloading it. There's tools in the MAST archive to be able to do that. We had a great time releasing the data, everybody, because over 6 months, the data wasn't public. And then suddenly astronomers wanted it all at once on July 14th, and they're just sitting there, show me the data. And so that created its own challenge. But within those first 5 days, MAST was able to deliver 260 terabytes of data around the world, 100 different countries. And this was possible because we got help from AWS S3 clouds. We put the data up there and let them distribute the data instead of everybody hitting our servers at MAST. So let me end just with one of my favorite beautiful pictures that have come out recently. This is the Tarantula Nebula. So now that you can look at this picture and hopefully appreciate what it takes to get to these beautiful imagery and how you might then also take spectra of the stars that are in it. This is another star forming region, but this one's like 160,000 light years away. And those stars just formed and are blowing away the gas around them. And so who knows what other treasures we'll find in our data archive as we continue to operate the web mission, and I'm happy to answer any questions.
There. I'll ask one as a enthusiastic observer of the papers. Rather than from the papers were reporting something about some mirrors being damaged by micrometers. You were updated somehow. I couldn't tell if the papers faster.
Yeah, I know the papers definitely made it sound really serious. We always expected the mirrors to get hit by micrometeorites. And in fact, those time series observations I talk about, you can actually see then when it happens. So we're talking specks of dust hit our mirror, and it does do a little bit of damage to the mirror, but that was always part of our calculations. The reason it came up is we got hit by one reasonably larger one, larger than we expected. And so it happened so fast, it made us rethink, like, do we have our models for how often we're going to hit these wrong? And so we wanted to go back and rethink it. And so we're now a little bit more scared about pointing straight into the direction of the velocity where these things come from, but it's not so bad that we won't do it. Like sometimes the science warrants that sort of risk. So, NASA's all about let's weigh the risk against the scientific reward. So it sounds terrible, like we're getting hit by meteorites, but it's not that bad at all. It's these tiny little dust particles, and over time they will degrade the mirror, and we're just hoping it doesn't happen too fast. Yeah.
Thanks for fascinating talk. Two questions. One is perhaps more on the mechanical engineering. And no one sees this thing kind of pointed one way with the base and the reflector. And I'm just curious.
You mean
we focus the entire, point in another direction. How is that actually done?
Let me
take the entire thing or this thing.
Okay, yeah. So the bottom has, you can't see this at all. There's essentially jets on either side. And so the whole observatory moves, but you're in space. So you're not running into fluids or anything like that. So, but they do. So the whole telescope was spec'd to only last for 5 to 6 years. And that was always based on how much fuel we ended up in there because Ariane gave us a perfect launch, and the last little bit comes from those jets. We only had to use a little bit of that fuel to get us into our L2 orbit. We actually expect to be able to last 10 to 20 years on that fuel if nothing else goes wrong, right?
Second question is, I always thought the galactic center was cloud in between and it's hard to observe. Is this going to help studies?
There are studies that are going to look at the galactic center. I know, for instance, that big Event Horizon telescope that did image the black hole at the center. There's a project to work with them to, I'm guessing, improve those images. It's not really my science to do that black hole in the center. So there are people who are looking at that. So the high resolution of Webb will help separate the images, and also because we're in the infrared, you can get through some of that gas and dust. But exactly what they're studying, I can't go into detail for you.
The data pipeline is interesting. I wonder how much was happening on board the spacecraft and how much is down to the level of numbers.
So the whole pipeline I'm showing you here is all in Baltimore or on your laptop. Very little, almost none, in terms of processing is going on board. Essentially, the detectors get read out and they're put on a hard drive.
I think I was next. Astronomy as a field has always been kind of characterized by deep involvement of hobbyists, managers, and other citizen scientists. So it's really exciting to hear about the release of all of these completed databases. I'm interested in if there's any plans, around to incorporate with that, like open source data model or open source analysis tools used by them to get a deeper insight into the data, that is kind of the raw information out there.
Thank you for that question. This pipeline is entirely public and open source. You can go download it and use it. That only takes you so far. It gets you to these perfectly calibrated images. We are also working at the Institute to have data analysis tools to do better visualization. So look up JDA-Viz, which is the one we're working on. But yeah, that is from mass perspective. I want everybody to have the data and have all the tools, and everything will be open source, and you'll know what is happening to your data. You are welcome.
I'm very excited about the first image published. And is that the raw build out image, or use like some time period image data and after filter and build out the image?
So this image here you're talking about? Yeah, but that is the first one. Okay. The very first one. Yeah, all of these images, I'll go to the very first one, sorry. As I said, all these, yeah. So this comes from averaging together a whole bunch of individual exposures, taken across 6 different filters. So you have to observe in all 6 filters. And then you say 2 of these filters are my red, 2 of these filters are my green, and 2 of these filters are my blue. This is why I wish I had my other talk, because I put this slide in. And then you average them together with those 3 color bands to get this color image. Then there's a lot of artistry that happens as well to get to this.
Is it like very strong commentary? Like
let's see.
Some aggregate value.
So you're asking how long the exposures were essentially? Yeah, so approximately it's like an hour per exposure. So this is a total of maybe 6 hours of light. I can't remember exactly what it is, but it's certainly not days. Like the previous deep image from Hubble that you probably saw was exposing for like a week. And so this gives you some sense of the just because we have such a big mirror, we can go so much deeper, so much faster.
I have to not be fascinating. I really shout that it is fascinating. So you mentioned 5 years. So I'm expecting another launch in 5 years. And you also mentioned the difference of the jump between web and the previous one. And the different days. So my question is about is the future for astronomy, and what is going to happen to all the land telescopes that are telescopic achievement? Is there enough space and science for everyone?
So is there enough space? I missed the end of that.
Space, stars, science.
Is the universe big enough for us to have all these telescopes? Is that what you're asking me? Yes. Each telescope kind of offers a new, unique way to look at the universe. Like, not all instruments can do exactly the same thing. Just because you have the biggest telescope doesn't mean it's the most ideal thing for doing your science. Here's a great example. TESS is a NASA Space Telescope that I work with a lot. And it's actually very small. It's 4 very small telescopes, like this big, but they have the ability to stare at a large picture bit of the sky. And as a result, they can look for those times when you see transits, like I was talking, from the planets passing in front of the star. But you couldn't do that with Webb, because Webb only can really look at one very tiny patch of sky at a time. So it's not a survey telescope, right? And so each telescope kind of offers its own unique look at the universe. So yes, we will have plenty of science to do. I am expecting the next call for proposals for Webb to give us, probably a 8-to-1 oversubscription rate in proposals for it. And Hubble is consistently sitting at 8-to-1 as well.
That's why, just a quick final thing. Is there anything that Webb can't do better than Hubble?
Well, it can't look at optical and UV light. Yeah. I'm a terrible person to ask, because I've not really worked a lot on Hubble data. Yes is the answer to that. But right now, those communities all looking at Webb right now, so I don't have a clear answer for you. I'll think for an hour and I'll give you a good one. I should have that answer.
Well,
don't feel bad. As I said, people will still really want to use Hubble just to have the full wavelength range, right? There's chemistry you can get in the optical and UV that you just can't do in the infrared.
Well, thank you so much. I was so excited for students talk and previous items. Let's see the funding on this. So there's 2 that we missed, and let's start with the first one. Strong Wolf, and every time I think Strong Wolf, I think on fire amenities. And of course, it's a different spelling, so it's unfair. Tom is a research professor in the Department of Physiology in the School of Medicine. He started the development of something called Daily 24. Project and Apple gives health kit research kit, and then the seed funding project is about COVID-24. So 24 is a theme. It's about COVID-24 now. 3-year application.
There's other applications. Yeah. Okay. Yeah. 3. Yeah. So well, it's a pleasure to be here. I want to thank everyone for staying on, for the afternoon session. I did note we had a difference in the populations between the morning and the afternoon. So thank you all for being here. And thank you all the organizers again for setting this up and for the seed funding. It's been tremendous for us. So an opening question, does anyone know what this is? Why did I bring it in? It's part of our project. Some of you may realize what it is a little bit later, because I'll talk about it. This will detect CO2. So one of the things that our proposals on is COVID, as I think everyone caught, because we caught it COVID-24. So hopefully you recognize what we meant by that. And one of the risk factors when you go to a public meeting is the number of people in the room, CO2. So you can buy these. This is one that we purchased as part of our application development. And I'll put up the room. You'll see in the middle part of the talk, we're below 800, which is kind of the magic number. If you get above 800, you kind of want to maybe ease towards the exit. But if you're below, you're fine. And we've been about 700 to 750 throughout most of the meetings so far, so it's been fine. But this is something that's not too expensive to get. If you're curious, I actually have been enjoying it just independently, even of our COVID-24, because you can measure things like radon if you're interested in that, VOC, things like that with these. So anyway, continuing on. So this is Metabolic Compass, and as was brought out earlier, we were one of the first awardees, I think it was second year, if I recall correctly, that I did started. And so if anyone knows John Ahmad or Jeannie Clark, Jeannie's head of general medicine here on the medical school campus. John is now Google, but he was in computer science. And I was a kind of guest member, so to speak, over computer science for a while, working on streaming databases at that time. So we were curious about timing and eating. So daily 24, I think you can see my mouse. No, you can't.
Okay, so daily 24, we were asking people to put in their timing of eating and their time of sleeping. And the app was addressing whether you could lose weight by paying attention to when you ate. There's a lot of animal work on that. And this is really, I think, one of the success stories of IDEAS is that we did get national funding for this. So this led to kind of our exit point almost from daily 24 was right around when COVID first started. We had 4 solid years of funding from the American Heart Association for what started from an IDEAS project. So again, we were incredibly grateful for that. And so like many people, we had this app, this is AWS backend, which I'll go through in a minute, to React Native on the front. So we released it. Well, actually, this one is Swift. The next one, Daily 24, is, you'll see in a moment, this Android and iOS. But like many people, when COVID hit, I was thinking, what can we do? What can we do to do something to help? And I could not come up with something near the beginning, but now near the end, we hope the end. We did come up with something. So this is daily 24, and it's a more user-friendly, I think you can see interface, but we don't return as much data. So what we have was people inputting again, things like their meals, when they slept, and then on the right, as you can see, we gave them a dashboard. And what we're doing in our revision is ultimately trying to help people think about their risk. And the problem with COVID, as most of you know, is that it's something that's gone up and down in terms of people's awareness. It's gone maybe up and down in terms of the number of hospitalized. I think it is going to become something that's going to be always with us now. I don't think it's going to completely go away. But we realized when we put the application in for IDEAS funding, that was in January. We thought there would be another streak of interest in a dashboard to predict risk, which is what our initial entry point of this was. As we got funded and as we got into the summer of development, we realized there's much more here, and we've actually submitted 2 NIH grants to try and get funding beyond this. But the idea is to look at long COVID, which is a chronic now disease, very similar to some of our entry point with daily 24, because that was looking at weight control and pre-diabetic metabolic syndrome. So we're looking at long COVID, and then we're also looking at acute respiratory distress syndrome, which I'll try and give you a sense of our 2 proposals that we did. So that's looking at lung function in RDS. There's other things besides just COVID that can lead to that. So I guess is there a pointer here? I'm so used to using a pointer, but no one can see. Okay, well, I'll just describe it then, and I apologize, but hopefully you can all see on the left, we've got a pre-exposure, then an incubation phase, then the viral symptom phase, inflammatory phase, and recovery phase. That's that top row with the disease phases. Then there are these mechanisms, you've got the viral replication, and then you've got the inflammation response. So this one, if you look on the x-axis, that's just the first 28 days. This is the most common for people who do have COVID. It only lasts at most 4 weeks if you don't have long COVID. And in particular, you can now, with the possible treatments like the antivirals, and especially with vaccines, obviously, you can prevent a lot of this from getting to the most extreme cases. So what we're interested in ultimately are now with our 2 layer versions of COVID-24, we're interested in this inflammation response, this slightly longer response, and then that would potentially turn into long COVID, which is this second slide on the disease progression. So, sorry, I don't have the right position on the mic. I keep modulating my volume. I apologize. But the question in long COVID is some individuals have symptoms that go well beyond 28 days. And what's going on? And it's not completely clear. It's not even in terms of the EHRs, the records that people put in. There's not a clear diagnosis criteria, but there's a huge amount of anecdotal evidence. So we're now even proposing something called medium COVID. There's a huge amount of evidence. This is a kind of scary, I would argue, but emerging trend. And the question will be how you treat these people, how you help these people. And so the first proposal we put together, which I'll put up in a moment, was really to try and help people with medium and long COVID. So you may not be able to see this too terribly well, but you can see there's weeks down along the bottom. And by the time you get past week 4, which is the 28 days, you're starting to get a major impact of COVID in other organ systems, not just in the lungs. Most of you probably know that the extreme versions of regular COVID, if they end up in the hospital, the acute respiratory distress syndrome, that's largely lung function. It really is just like an extreme pneumonia. I mean, they're not getting enough oxygen. Long COVID seems to be, and maybe medium COVID, these other organ systems that are impacted. And right now, a lot of the evidence there has been collected through surveys. There's not a huge amount of direct clinical information that's been collected, but I think there's going to be a major need in things like general medicine, just put in the proposal with for Green Spring Station, as we where Green Spring is to compare against other stations. The question will be, can we collect enough information on some of these post-4-week symptoms to try and help with diagnosis and with treatment? The other thing we're using is another Hopkins success story. So we know this is N3C. Talk about huge databases, maybe not at the scale of the web, but really large. So hopefully you can see some of the numbers: 12.3 billion rows, 1 billion clinical observations, 11 million people, 3 million some COVID cases. This is an open database sponsored partly by NIH, but led largely by Chou, one of the people you saw, involved with the several things related to our IDEIES program, and very involved with informatics at Hopkins. So this N3C for National COVID Collaboratory 3Cs has been instrumental for helping to see things. And this is kind of our backbone reference data for comparison with all the data we hope to collect from individuals. And just to give you some idea of the architecture, and if people are curious, maybe offline don't have a large amount of time, I can go through more of our architecture. But right now we're building the new version, not yet fully released. We're trying to make 3 versions kind of in one. But it's, again, as I say, a React Native app with AWS on the back end. We're using Postgres data stores. We use S3, and especially things like Spark, which came up as well, for here in town. And then we give feedback to the patients. And so in particular, we've got wearables, and that's this. Right? So this thing is AirThings. If anyone's curious, you want to look it up, you can buy it from Amazon. Essentially, it'll measure things. You can see there's an app on the right. So you can measure all sorts of interesting things about your home air or the air in the environment that you're in. For COVID concern in the meeting, obviously the concern is this CO2 level. And essentially, this is something that Mark pointed out. She's very well known, Lindsay Barr, for her work on aerosol spread of COVID. And essentially, what you're looking for is whether you cross a magic number. That's 800. There you go, right? Thank you very much. So whether you cross this magic number of 800 is the concern. So what she suggested, and there's been other people suggesting that too, is if you really are concerned, and I think in some ways there's been less concern about it than I expected, but if you were concerned about COVID and you're going to a meeting, you do what I did today, you bring your CO2 meter in and you monitor it. And you say, how good is the room? And we've got big ceilings in this room and we're not totally packed in here. But if, for example, I had come in, I was relieved to see it wasn't this way. But if I had come in and there were 200 people in a room this size and we didn't have good room ventilation, I might not have stayed, right? I would have suggested other people, do something similar. But that's only if you're really, really concerned again about COVID risk. And again, for us to evaluate COVID risk in the app, what we're doing is taking, and the data here is less available than it was before, the CDC or our own web app here at Hopkins, based on zip code, you get some idea of the COVID release in the surroundings. And so risk is then just a fairly easy statistic, number of people in the room, number of minutes or hours you've been exposed, and rough percentage of those people that might be carrying COVID based on the community spread. So it's not a hard number to compute on our back end and put back on people's apps. So this is what I said we would do. It's just more curiosity. So from home, you can see the average was 462. Here in the meeting, you can see on the right, 767. So we're below, that was just around lunchtime. So we're below 800. We can all stay, finish up the meeting, no huge risk, especially those of you that have masks. I think it's lower risk. So returning to this and how we calculate and what sorts of things are going on, and especially to our grants. And the second grant I mentioned, ARDS. This is with Grant Gerard, who's been very involved with treating COVID and isolation units here at the medical school. What we're interested in doing is from CT scans. Many of you know this kind of radiology. You can actually reconstruct the lung, and you can actually make very interesting fluid flow type models, airflow models for the lung structural function. And the idea will be that we can put this on people's phones. This is part of the app. We're still in process on this. And then we're supposing they'll get a wearable, like that thing you can see in the middle. That wearable basically is like a digital oscillator. You can hear through it, right? Stethoscope. And those locations, you can see those 6 on the back, 3 in front locations. You would know from the CT scan. The idea would be to collect people early. The idea would be you help people see where potentially they've got a lung infection. And so this was proposed to NIH, and Joe in the back there helped me put it in. So submitted through IDEAS. And this went in just 6th of October as a proposal. This one was actually the first one that went in. And the idea is to connect it to EPIC. So the original one, I'm getting close to it again. The original idea was to, as I said, evaluate risk. But what we realized is that we're going to be seeing a lot of people with medium and long COVID, and so we want an ability to monitor them. So this is Green Spring Station, and the idea is we'll connect it to EPIC and help people, we hope, see their risk. And so these are just a few bits of our risk analysis. This is a 3, 6, and 12-hour mark. This is based on patients from an ICU setting. It's not exactly the same, but testing our software. And you can see that risk changes as a function of time, and that's the general idea we want to get at. And on the bottom right, there's maybe not time to absorb it, but pneumonia patients also from the ICU data set change the risk changes with circadian status, which I thought was quite interesting. So this was the title that we put in. You can see there's a number of people with me, and I just wanted to acknowledge and thank all of them for contributing. It's been really a team effort from all the people that are involved. And again, it was originally just risk outside of clinical setting, and now we've added in clinical setting. So with that, I'm going to stop and say thank you.
Thank you, John. Quick question. Just to clarify, COVID 24 versus 24 hours, not 2024.
Correct. Yeah. So daily 24 is 24 hours. We just took the name COVID 24. Yeah, exactly. But if it was COVID the year 24, we'd be concerned too. Yeah, thank you.
So a quick question about the data flow you showed in the AWS.
Yeah.
And that comes from the app. Did you have to deal with any kind of compliance issues?
Yeah. I didn't really mention it, but this is with data. I think I mentioned it on one of the questions at one point as well. So we have to be very concerned about HIPAA type things. For daily 24, we were less concerned about it. But once we start bringing in EPIC, yeah, absolutely.
All right, thanks again.
Thank you.
Let's have another update from Meneveau. This is Associate Professor of Otolaryngology, etc.
Otolaryngology.
Sorry?
Otolaryngology.
Otolaryngology.
Close.
So she deals with comparative models of hearing loss, as well as the feedback in the ear and the brain. And this particular project is about the surgical database of read the rest.
Trying to turn on the laser. Great, thank you. Hi, everybody. Thanks so much for inviting me to be here. This symposium has been really useful for me because I think I come from a very different background from most of you. So this is making me think about our work in a slightly different way. Just a little bit of background. I come from an otolaryngology department, and that just means ear, nose, and throat. But I'm not an otolaryngologist. I'm a basic scientist in the department, collaborating with surgeons in the department who study the ear. And our department actually has been doing this for over 100 years, about 100 years, I should say. Dr. Samuel Crowe, this guy over here, actually founded the first otological research laboratory in the United States here at Hopkins to study deafness and hearing. And he actually was hanging out with this guy back here, William Halstead, quite a lot. And we did some investigation into the history of this this laboratory because we have a very large human temporal bone collection that resulted from it. It's one of the largest in the United States. What is the human temporal bone? I'll show you some pictures in a minute, but that's just the inner ear from humans. This is really the only way that you can study the cells that are inside the inner ear to understand the status of the ear in great detail, because you can't really image it with a lot of resolution using MRI or CT or something like that. So this is a very valuable collection that we have here at Hopkins from the work of Samuel Crowe. So he and his colleagues were spending quite a lot of money at that time. It was the equivalent of today's $13 million to harvest temporal bones from patients at the hospitals here and at Bayview.
and that this work was then followed up by George Nagger after World War II interruption. So this work was actually really pioneering at the time. What they would do is they would take the audiometer, which you see right here. This is the very first, one of the first models of an audiometer. And it's about as big as this podium. They would wheel this thing around the hospital and test patients' hearing. And this was actually revolutionary at the time because the way you would test people's hearing sensitivity was basically with a tuning fork up until this point. So this was high tech in 1924. And they would record hearing thresholds. Right here you see an example of these handwritten audiograms. So probably most of you have had some kind of test like this in your life, maybe when you were kids at the doctor's office. They put headphones on and asked you to push a button or raise your hand if you heard a sound until it was so quiet you couldn't hear it. That's all this is testing, right? Really, about 100 years later, audiometry still works pretty much the same way as it did back in the 1920s. But this was really revolutionary because they were doing this systematic characterization of hearing and deafness in a huge cohort of people. So they actually did this in about 20,000 different people, we think, based on the records. And they used this to describe patterns of age-related hearing loss that occur in humans, and made some note of things that are still not completely well understood as far as the mechanisms, but things like aging white males are particularly susceptible to age-related hearing loss compared to other demographic groups. So this is a human temporal bone right here that's been removed from the skull. And what Crow and his colleagues did was develop the way to cut these things into sections so that you could see in great detail the cells. These are just the sections. This is what it looks like if you take a picture with a microscope of a stained section. And this is a cross section right here through your cochlea, so the part that allows you to hear. They did this with about 1,800 specimens, and then stopped for World War II, did it with another 300 specimens. And what's especially unique about our collection here at Hopkins is that, because this was done in Baltimore, we have greater representation of specimens from individuals of Black or African American background. And this is kind of key because it actually turns out that people who are Black or African American tend to be less susceptible to age-related hearing loss. We don't really understand the mechanisms, but we think that there's something protective about the pigmentation that's in the ear that protects against hearing loss. So these guys, Crow hired and his colleagues, Stacy Guild and Mary Hardy, actually developed ways to quantify the cells in the inner ear, the status of the different cell types. And I won't go into great detail about these data, but this is what their data notebook looks like. Basically, they just mapped damage patterns along the coil of the inner ear, the cochlea. And their procedures for doing this really are still state of the art. So in 100 years, the way to analyze the patterns of damage in your inner ear haven't really changed substantially, right? And the problem is this takes forever, right? So you have to sit there at a microscope and look at many different sections through this ear and hand record damage patterns, right? This is, you might, if you're lucky, have an actual digital picture of that section instead of the microscope, but for some things you can't really resolve it without focusing in and out with the microscope. And you can imagine that's slow, laborious, and not a lot of people want to sit down and do that. So this is just showing the collection in the hallway, collecting dust since the 1950s for the most part, because people don't really want to deal with this laborious and slow data collection process. And to make it even more complicated, you have a whole bunch of 2D sections that you then have to try to figure out what that means in terms of a 3D structure. And ways to do that really haven't been well developed. So we proposed to make this a little bit easier and make this more accessible for people. So one of the big problems is that if you get digital images of these sections through the ear, you have huge data sets, and so sharing them is difficult. And this is where the IDEIES grant came in. So we want to just develop a proof of concept that we can build a database of these image sets so that researchers can access them and build tools to analyze these things in 3D and really bring analysis of human temporal bone tissue into the current millennium, because nothing has changed for 100 years. I think this is going to accelerate our understanding of diseases of the inner ear, as well as things like how it changes throughout development. And it's also going to be a great teaching tool because we have a lot of learners who are interested in this across the career spectrum, PhDs, MDs. It's going to be a great tool for learning and collaboration as well. So just an example of what that means. We have been trying to figure out the mechanisms under something called Meniere's disease for a very long time, over 100 years. And this is really kind of a mysterious disease. What happens is you have, when you have this, you have fluctuating hearing loss, and you have episodes of vertigo, very extreme vertigo. So it's really very unpleasant for people who have this. And basically, the most common treatment is to tell people to reduce their salt intake in their diet. There are some surgical manipulations that used to happen that were very severe and could leave someone completely deaf because they would just cut the nerve to the ear. And you can also inject the ear with gentamicin, which is an antibiotic that's actually toxic to the sensory cells in the ear. So none of these things are great, and they're not really based on a thorough understanding of the disease because it's so complicated to get information from these inner ear specimens from humans. But we've been working with a team to try to figure out some of the mechanisms that are responsible for these symptoms. And I won't go into great detail about what those mechanisms are. I just want to show you that our aims are to do things like take these 2D images and look at specific structures and see how they might be abnormal and rebuild that in 3D. And we want to do that in more than 2 ears, which this is one example of a study where basically they used 2 ears, 2 people, 2 ears to rebuild some of the inner ear structures to look at the volumes and how those might be different in Meniere's versus normal patients. So you can imagine 2 ears is not a very representative sample, but this is like years worth of work, right? So we want to accelerate that. Another example of why this would be useful is because there are parts of the ear that we don't understand the function at all. Thank you. There's something called the bast valve, which is doing something, and we don't know it. But we're currently using this to build 3D models so that we can apply what we know about this to both his surgical treatment approaches and also integrating some fluid dynamic models and experiments with that. So again, this is all done on a small scale. We want to do this on a larger scale and for other inner ear disorders. And for that, we need everybody to have access to this data in our field who wants to look at it. So this is a lot of work. We have to first go through all these paper records from these patients and catalog our boxes full of tissue. I won't explain the complicated logistics of setting up a digitization scheme, but this is something that seems like it should be simpler than receiving data from a space telescope, but based on the number of problems we had getting this going and getting this thing installed, I'm not so sure anymore, but it's working. This is our digital slide scanner with a robotic loading arm, which greatly accelerates our data acquisition for generating these images of sections. But now we've got about 1 to 2 terabytes of data per ear to deal with. And so our current issue is optimizing a workflow for transferring all of this data to SciServer, where we want to integrate some tools that can be useful for rebuilding these things in 3D and doing some more sophisticated analysis than just looking at single sections through these structures. So there's just an example of one that has not been manipulated through SciServer. This is just a master's student in my colleague Chellappa and Anathar's lab, who basically we had to downsample all of our images for 1 year. And it's been months that she's been trying to sort out how to work with these things and segment the different fluid-filled areas of the ear to try to build these in 3D. So we are hoping we can developed some tools to make this a lot faster in collaboration with their lab and some other labs using SciServer as a platform for sharing and accessing the data. And then lastly, once we are making this work, we want to scale that up. So we're going to do this with something like 10 years to start, but we have 2,000 years, something like that. So we want to eventually share most of those 2,000 years with the larger scientific community so that they can all tap into this and look at these data. So with that, I'll just say thank you to IDEIES for the seed grant. And I'll just note that we just recently were awarded an NIH grant to help further this work. And so we will be hopefully scaling up our efforts in the coming months and years. Thanks.
Time for a couple of questions. Okay. Well, thanks again. So our last seat funding of the day for today is from IDIES Executive Committee member, Paula Mancy. She is a professor and inaugural head of the Department of Chemical and Biomedical Engineering at Hopkins. And he also chaired the OSAC Committee for ARCH, Future for the Bottom Line. And she's going to be talking about seed project on active learning.
Thank you. Sorry, using somebody else's machine is always a little tricky. Okay, thank you very much for listening to my talk, and I want to thank the committee for funding this project. I was really delighted. I've only been at Johns Hopkins for the last 4 years, and I'm just still feel like, thanks to COVID, finding my way around. And so it was wonderful to get involved with IDIES, a real pleasure. So I want to talk about something a little different. And I mean, I think we've heard some incredibly diverse projects, and I'm going to also increase the diversity because I want to talk to you about manufacturing. And what we've been trying to do is to model the way that an additive manufacturing process works. So this is a project that has been done in collaboration with a couple of folks at APL, Dr. Namla, and also Gerard Gagnon. And what they've been developing is a novel method to what is called additive manufacturing. In other words, instead of etching something away and making a pattern and then laying something down on that pattern, that you actually add layers. And in this case, it's very unusual because what they've done is to form little gallium liquid droplets on a surface, cover it with an ammonia gas under a little bit of pressure. And then what happens is that the nitrogen atoms at high temperatures dissociates from the hydrogen, and they diffuse into the gallium, react with the gallium, and form gallium nitride, which is a high-value product. So that's what we're trying to model. So how can you do that? The most challenging part is the whole diffusion and reaction part. So if you were doing molecular dynamics and you wanted to do it on a non-reactive system, you'd have plenty of choices. So you can choose all kinds of semi-empirical force fields that will tell you what the forces are, then you can solve the F equals ma equation, and you can create a movie of how the atoms move in space and whether they would absorb, crystallize, or whatever they want to do under the physics. But they can't capture the reaction. And their accuracy for a reactive system is therefore a little bit suspect. So that's really out. You could look at one of the few reactive systems to create a force field that allows you to form and break bonds, but they're very few and far between. They tend to be very difficult to use. They involve hundreds of parameters. And they're not transferable to other systems. So it means that you've got to work from scratch every time that you add another component. So is there another alternative? Yes, you can use quantum mechanics. You can do an ab initio simulation, but the problem there is then that you limit your scope. You have a budget of maybe 200 atoms to work with, and you can follow that for about 10 picoseconds. That's not very much time, and it's not very big. And if you're trying to capture crystallization, it's not appropriate. So in this seed, we decided that we would use what is called on-the-fly active learning, which literally means that you're making decisions about whether or not to kind of call home to the mothership and get an accurate calculation from density functional theory, or whether you feel like, no, I got this. I've learned enough. I can make the next iteration on my own. And we used a model called FLAIR that was developed by Boris Kaczynski's group at Harvard. And it came out actually sort of, pretty much mid-pandemic. And when we saw it, we thought this is an exciting potential idea. And it could inherently capture reactions and diffusions, but it had never been done. In fact, I think that we really helped the Harvard group it was really at a sort of beta test version when we got involved. And so we tried to help they had some very simple toy problems, but we wanted to really extend it. So the key enticement of why should we use FLAIR is that we could get the best of both worlds. It's sort of like the holy grail for molecular dynamics people. You can get the accuracy of an ab initio quantum mechanical approach with the speed and the scope of a traditional molecular dynamics. But we didn't know going into this whether FLAIR was capable of capturing both the diffusion and the reaction of the system. So the way it works is that you essentially use, at first, you make a few density functional theory calls. They're expensive, but at least it gives you a very good starting point for moving your simulation along. So you can then initialize the Gaussian process. And here, unlike a lot of the other talks, we're not dealing with big data, we're dealing with little data. So our approach is much more a Bayesian optimization, which is really good at studying systems where you have expensive and sparse data. You predict the forces using the Gaussian process and your surrogate model, and then you update the position. So you move like a frame in the movie. So all the particles readjust based on the forces you just calculated. Then the key step is you check the uncertainty of that prediction. If the uncertainty is low, you say, okay, my learning is good and I'll keep going. If the uncertainty is high, then you have to call back and make another density functional theory calculation. So obviously the trick here is to make as few density functional theory calculations as you can and go merrily learning along the way if that's possible. So what our tasks were to train the model. And one of the first things that we had to deal with was the fact that we've got all three major phases of matter: gases, the ammonia; liquids, the gallium droplets; and hopefully creating gallium nitride as a crystal. So we had to give it a little bit of information about each one of those. So we made some runs that determined what ammonia would look like and how that would dissociate. We studied bulk gallium nitride in the right crystal structure that we thought should thermodynamically be stable. And we also pre-trained on ab initio molecular dynamics. And that's one of the things that we did that the original Flare developers did not do, because we found that it made the training go a lot faster. Then, of course, you have to validate. So we just pulled as much experimental data as we could find, and then use that to calculate properties that were in the literature, and that we could then say whether our model was decent or not. And the hope on this is that we'll now be able to tackle more difficult problems that the experimentalists can't see. For example, they can't watch the growing crystal front as it moves through the melt, through those liquid gallium droplets. And you want to know which facets are moving the fastest, because that affects what you eventually grow. It affects what defects you create, and hence the quality of your material. So the critical thing is, can we get this thing to crystallize into gallium nitride from the melt? So we did quite a bit of validation, and it's hard to see, but the leftmost panel is showing you what happens as a function of time as you're raising the temperature. So as you raise the temperature, the blue points are saying all the learning points, and the little red crosses are saying every time that you had to make a density functional theory calculation. So what tends to happen is that as it's moving along, you tend not to have to call density functional theory at all. But when you get to something maybe more catastrophic, like the system melting or the system crystallizing, then all of a sudden the uncertainty will go up and you have to make calls. One of the things that we track is the middle panel, which tells you essentially the error. And we have, it's really kind of like a rule of thumb of how much error we'll tolerate. But we also know that that's sort of a necessary condition, but it's not sufficient. Just because you've got a low error doesn't mean to say that, it just means it's kind of being self-consistent. We also calculated properties that we could get experimentally, and I'm not asking you to read them, but if you can take my word for it, because I want to get on to the most important thing. The prediction of bulk properties was great, but it wasn't our chief focus. Our chief focus is this slide, and that is when we have a melt, and so that's the disordered parts on the left panel, where you can see there's no order, and in the middle there's a very ordered structure. So we put that little seed of work site structured gallium nitride, and what we're asking the system to do is to grow off of that. Now, how do you find out whether it's actually growing a crystal or not? Or is it just getting other gallium and nitrogen atoms just getting close to one another? So in order to do that, you need to invoke the idea of an order parameter, because in a crystal, you expect all the neighbors to be at very specific angles and distances. And so that's what this is doing. And you see 3 panels taken at time t=0, after 4 nanoseconds, after 8 nanoseconds. And what you see is these little green X's tell you for each of the sort of layers, if you bin the molten parts by layer, and you plot their order parameters, there are 2 order parameters, Q4 and Q6. And where we're going is we want to get to that red star, because the red star is what the structure should be if we're really growing gallium nitride. So what you see is as time goes on, the little green symbols kind of march their way up towards that red star. So what that's telling us is that order is evolving actually pretty quickly after only 8 nanoseconds towards the point that we expected it to do. So that really pretty much categorically proves that we are growing work site crystal. And I have to say an important point here, which is that my student believes, and hence, of course, I believe, that we're not biasing the system to form a work site structure, but we are actually going to test that in the coming days. So to conclude, we've got a new model for the creation of gallium nitride has no parameters. It's fast. It's a big system. It allows us to really watch crystallization. And we've also found a few small things that will improve how people who use FLAIR will perhaps model things in the future. Now, all of these on-the-fly models are pretty fragile. So I'm not endorsing FLAIR per se, but I'm saying that my student did a heroic job in getting this to work. The products that we made, we've got a paper that is under revision at the Nature Publishing Group's Computational Science. And we have a proposal that we're preparing in order to fund this work. So with that, I'd like to thank my collaborator, Dr. Namla, and of course, the person who really did all the work, and that is my great student, Aaron Chen, and of course, IDIES for funding, and Arch for providing us with lots of cycles to do so. Thank you. Hi.
Which type of was selected by these business? When I use that, for some of this program is something explanation does not work, which I have to use less in.
Yes, yes, we were actually testing different kernels, but what we typically have a range now of maybe 3 or 4 different kernels that we try, and we're checking to see that all of them actually are producing much of the same thing. But certainly, like we start with sort of naive beliefs, like just one hot encoded, but we're moving now to a property-based one, which we found is better, and it keeps the physics in there. As for lamps, I think you mentioned lamps too, right? Matern. Yes, yes, that's what we use. Yes.
In some case, I have my very sequence. So when we're talking about a method and highly involved, we're talking about cognitive therapy. But the method is more like a method, so we'll have to get to call the 2 variables. So I quite understand how we control it.
Okay. Yeah, I mean, so what we did in that situation was just to simulate ammonia and then take it to very high temperatures. So it's like, yeah.
The density functional calculations, are they done on the entire system or just on a few particles?
Yeah, they're done on the entire system. Yes.
Sometimes you can get away with finer grain, more accurate on subset.
Yes. And that's certainly what we did for the ab initio. So that's all very small scale. And that's put into the training set. Thank you.
So, okay, thanks again. I believe you're also presenting the Robin.
I am. So probably the best part of my job on the executive committee for Arch is to run the Robbins Prize. And I get only the fun parts. I don't evaluate. In fact, when we set up the rules, it was made clear that I would not look at any of the applications. So I get all the wonderful parts, which is to announce the winners. This is our 2nd year of operation. And this, of course, was to commemorate Mark Robbins and his incredible and lasting contribution to high-performance computing at Hopkins. And so we're very pleased to have this started. And I think it may be the only high-performance computing award that Hopkins gives itself. So it's very special. And there's 2 kinds of awards. One is the Future Faculty Award, and that is given to a postdoc. And the nice thing about the postdoc award is that you get some funding for computer time that is outside what your PI, your postdoc advisor, actually gives you to work on. So you can start to develop some independence and become that faculty of the future. And so we're really delighted that this year it was won by Dr. David Buchter. But he's not able to be here, I'm afraid, but we should give him a round of applause. And Dr. Buchter is helping to advance computational fluid dynamics in his work. There's also 2 graduate student awards. One of them was given to Samantha Zarate, who works with Michael Schatz on computational genomics, but I don't think she's here either, right? It's unfortunate, but let's give her a round. But delightfully, thank you. We do have one of the 2 PhD awardees, Dr. Remy Yovanov, who works with Albert Lau, doing all atom molecular dynamics and looking at computational biophysics. So, Remy? Please. It's quite heavy too, isn't it? Oh, so I get out of the way. All right, let's get ready.
So I just want to add my undergraduate degree in all the reports. First of all, I'm so glad there were so many of them on the TBA category reports. And second, they were amazing. They were really, really good. Thank you to everyone who submitted the proposal. We only have a limited number of awards, but they were really great. Thank you.
Our last talk, and it's a keynote, is a remote talk. So the speaker is from the West Coast, Gerard. He's a software architect with Salesforce. And
yeah, and we're gonna.
So he can like distributed database systems and also high performance computing systems and multi-process with over 40 years. And he has very deep roots in database technology, including transaction monitoring and fault tolerance SQL. And his talk title today is Standing on the Distributed Shoulders of Giants.
One second. We don't want to share. Okay, stop video. It's supposed to be sharing from his. One moment.
You need to unmute first.
How's that? Can you hear me now?
Yeah.
Okay, let's figure out how to get the screen shared. Nope, still disabled.
I don't know. Stop video. It says you're allowed to share.
Perhaps it's the other account. Right now I'm disabled.
Oh, okay. Okay, let me see. Let's try this one. Or video. No.
Stop that.
I need to know how to get the.
What do we need to do?
We need to get him allowed to share.
With the other laptop with my name, not the one with the video on.
Oh,
no. Okay. We can use.
Logged in twice. One will be the slides and the other will be my face.
Can you just allow everyone, Kate? allow everyone to share.
Allow everyone to share. Okay.
Yeah, I think that's the easiest.
Still can't share.
Oh, hang on, share. Oops. Okay, I think I set multiple participants to share, so.
Okay, we're getting good. Desktop. This should be good. Now switch to the PowerPoint. That looks like what I wanted.
Okay. And can we spin the computer around so he can possibly see us? I want to see if that
I'd
works.
be great to see the room. That would be awesome. So I'm Pat Holland. I'm very grateful to Alex Saleh for inviting me. And I've been doing distributed systems since 1978. I honestly had hair back at the time I started doing distributed systems and database stuff. And I tend to write papers for ECMQ and other random things with just crazy ideas. And Alex asked me to do this talk. It's called Standing on the Distributed Shoulders of Giants, Understanding Distributed Computing by Looking at the Great Physicists of Yore. So I do work at Salesforce, but these are my ideas. They're not necessarily Salesforce's ideas. That's great. I can see the room. Thank you. That's better. So let's get started. And interrupt me if there's anything you want to ask about. So this is a paper that I wrote in 2016 for communications of the ACM called Standing on the Distributed Shoulders of Giants. And so if you want to go look at that, that's also something that can happen. And the idea is we can use the great physicists and their innovations as a vehicle to help explain distributed computing. And I'm going to go over 5 great, physicists from the past. There's Newton, who is, 1642 to 1727, Einstein, 1879 to 1955, Hubble, of course, Werner Heisenberg, and Erwin Schrödinger. And then we'll conclude.
Oh,
I don't have to
That's a problem.
Pat, can you stop for a second?
Sure.
Can't see the slides, unfortunately, here in the room.
Oh, no.
Yeah, we're still having.
It's not getting input.
Yeah, maybe I can put it in there.
So the projector's not carrying forward what Zoom has?
That's looking better.
Okay.
Just hold on a second.
Can you see the title slide?
Yeah, there we go. All right.
All right. So the first thing I wanted to say is the content of this is out in a paper in Communications of the ACM from 2016 by the same name. And the broad idea that I'm going to talk about is using the great physicists and their innovations just as a vehicle to explain distributed computing. Then we're going to cover Newton and Einstein, Hubble, Heisenberg, and Schrödinger, and talk about how their kind of popular perspective of what their ideas are can be used to explain distributed computing. Let's start by looking at Newton. Newton thought he knew what time it was, man. He was a bad dude. He did classical mechanics, laws of motion, universal gravitation. He had the first refracting telescope, developed a theory of color, and much, much more. Like within a single computer, like a von Neumann computer or a small number of close ones, you could have transactions or a clear notion of time. You had only one time, and then each transaction follows others, time marches on. One thing happens at a time, and there was kind of one notion of time in a Newtonian universe. You didn't think about time being different across the space. Now, I studied computer science when Nixon was president. We didn't think about computers talking to other computers because you were really lucky to have one and nobody had two. And once in a while, there was a tape that came from some other, computer facility or something, but we didn't think about time across computers for a number of years. Now, let's look at what Einstein kind of said. So one of the things that Einstein said is, he kind of had many watches. He said time's different in different places. And he was amazing. In 1905, he published 4 brilliant papers: photoelectric, Brownian motion, special relativity, mass energy equivalence. And special relativity is kind of what I'm going to focus in on. He did so many other contributions. But he said all laws of physics, including time, appear the same to all observers. And what that meant was the speed of light is changing, but time was not changing. But it also meant that your time and my time are not necessarily the same. And that's a complete, twist of the mind when you think about that. And there's interesting consequences of special relativity. The sun might have blown up 5 minutes ago and we won't know about it for 3 minutes. It's going to be a fine 3 minutes till we find out. In computing, it's similar. when stuff happens far away, it takes a long time to find out. So that's something deep in this environment, is to say, look, computers are not quickly tied to other computers. Things that the other computer does is a long time ago, and it may take time to figure it out. And so in computing, you actually can't know what's happening over there. What you know is I can send the message and then wait for some answers. Latency is a big design point. But in distributed computing, we can't really know how long it's going to take, a little bit unlike Einsteinian physics, because the network has queues. All of these things make it irregular, but it's clear that you and the other computer aren't in the same time world. Now, in an Einstein's world, it's hard to get in the last word. You can't know if a message has arrived within bounded time. It's all probabilities, and the longer you wait, the more likely it is. But you can't know about that last message because you have to send another last message. So if you look at the left-hand side here, the server A talking to server B, that final message surrounded in the orange bubble, it's not guaranteed. You never know what got there, and you can't know. And similarly, there might be two last messages in the same order, and you can't know. In a request response, you can know the request got there because the response told you, but the server B doesn't know if the response got there. And then, of course, there's fire and forget, where you're just spewing out a message and you don't even know if it got there. So the sender of a last message cannot know it arrived. You only know when you hear back, right? That would take another message, so that's not the last message. Messages in servers, they have their own time. Servers can stall, messages get lost, response to the first message may be the last to arrive. So in the picture on the bottom, server A sends a message to server C, and that's the first one that leaves server A. And then it sends a message to server B, which is the second one leaving server B's time. And then the one can go from A to C, and C sees it in its time, and then server C sends it back to B. And the ordering at B may or may not be causally related in timing to the order that A sent them, because these things are different in the way they arrive into the different things, because things can go slow and things can go fast. And it also turns out that this is part of why you can't do distributed consensus in bounded time, because messages probably get there. You can't know when they all get there. And so things like Paxos and Zookeeper and Raft, they can take forever. They're likely to reach agreement soon, but there's no guarantee. Each server in its own world doesn't know what's happening over there. And some of you may have heard of the CAP theorem, consistency, availability, and partition tolerance. And the idea behind this is, if you look at those three properties, consistency, availability, partition tolerance, and here consistency is classic read-write, reading of values, writing of values. If you pick, you get to pick any two of those, but you can't pick three. And that was a conjecture postulated in 2000 by Eric Brewer, and it was proved in 2002. But I like to point out that that proof only applies to consistent reads. It doesn't apply to things like, oh, enqueue my email message, or other things besides reads. And so we get a little tangled up on exactly what this means. And because it is only applicable to consistent reads and it causes all of this confusion, I'm kind of fond of saying that you get consistent reads, availability, partition tolerance, pick two. CAP is really crap. And so it's very confusing in this whole world of what you can and can't do when you're disconnected. And also two-phase commit, which I shipped the second one in the world at Tandem, it was around 1982. It provides strongly consistent transactions, but it does so by deliberately sacrificing availability to make sure it can give you those consistent transactions. And so I'm fond of saying that two-phase commit is the anti-availability protocol, which guarantees consistency over availability. So we're making good time. Does anybody have any questions? Anybody think I'm full of craziness at this point? Are you guys awake? Okay, well, we'll keep going. Let's talk about Hubble. Okay, he was really cool. I like to say Hubble was increasingly far out. Edwin Hubble learned the universe is expanding. The farther away an object is, faster it is receding from us. And everything is getting farther away from everything else. Now, In our computing world, we see expanding computation bandwidth and memory. Even in Moore's law is crazy. We keep getting twice as much stuff every 18 months or so, and the bandwidth is going up even faster, but the latency is not dropping as much, okay? Latency is limited by the speed of light, and I'm not sure we'll communicate faster than the speed of light very soon, but I'm not holding my breath. So while you're waiting for an answer from something far away, because you're able to think more and more per second, you actually lose more opportunities to think as you wait for answers. And so the act of talking at a distance is becoming increasingly expensive, and it's been doing that for decades, and looks like it will continue to be the case. And so I like to say computing is like Hubble's universe. Everything is getting farther away from everything else. The number of instruction opportunities that are lost while you're waiting for a DRAM reference for your local memory continues to rise. I mean, I recently did a calculation, and compared to numbers from 1995, it's something like 20 to 50 times, depends upon your instructions per cycle, when you look at the fact that DRAM continues to be 80 nanoseconds to get to, but you can do more computing in those 80 nanoseconds than you could before. So the pain of a cache miss continues to rise. The pain of an RPC continues to rise. Anytime you talk at a distance, it's getting more and more painful over time. And to me, that's a lot like Hubble's universe. Now, I'm going to say I don't think Hubble didn't share very well, because as stuff recedes, sharing read-only data is not too hard. You use caching, it's immutable. If it's immutable, it's a whole different world. Everybody can look at different copies of the King James Bible without getting too worried about coordinating. If, on the other hand, you're sharing writable stuff where you're trying to modify things in shared memory, that just gets worse and worse over time, because you're stalling to pull the latest data from somebody else who's modifying that shared memory, and you get more and more instructions lost while you're waiting, and setting a semaphore in order to control who gets it gets worse and worse as time goes on. And so, shared memory where you're writing, it only gets worse as time goes on. So I like to say that shared memory works great as long as you don't share memory, because it's hard when you have to really share it. And so we need to do something about this in our designs. And we either are going to get around that pesky speed of light thing, or we're going to work harder on immutability, asynchrony, and concurrency. Let's look at Heisenberg. This is a smart dude. He defined the uncertainty principle. The more you know about the location of a particle, the less you can know about its motion. Basically, you can't know everything about anything. And in distributed systems, you can have a gaggle of servers. There's a bunch of them, you're working together. But they're in various states of health, death, or garbage collection. And so they're not always going to get back to you quickly. Most of the time, you get a quick and crisp answer, but sometimes they're just kind of taking a coffee break or on vacation in Mexico. It could take a long time. When should you give up? Whenever you're talking to a single server, you don't know when you will be delayed. So frequently, the response time is very important. Like I work on building database systems and people are waiting, and the logging time is super critical, right? And so you can launch multiple requests. And if you can wait for a subset of them to finish, then your probability curves just went very different, because I can get a certain number, I can get a quorum of those responses with a pretty predictable behavior, even if 1 or 2 or 5 of them are going slow. If you don't care where the work happens, you just care when it happens. Now, one of my favorite papers of all time is The Tail at Scale by Jeff Dean and Luis Andre Barroso at Google. And it's about managing latency at Google, and it's CACM February 2013. And they're doing read-only requests, specifically a portion of a search. I'm doing a Google web search. I've got to get to 1,000 different shards to see what kind of documents match this pattern. And it's idempotent, because it's okay if I do it twice. I may get a slightly different answer because that particular replica of that data might be slightly different, but it's okay, it's all good. And there's a natural variability in the service request timing. I talk to that server, and mostly they're pretty fast, but once in a while they're tangled up with things like garbage collection and maintenance activities and queuing. And so sometimes the requests are slow. And so what they do in this paper, and this is how Google was so fast, is they retry each request after reaching the 95th percentile. So they know how long a typical request is expected to take. They wait until 19 out of 20 of them would have answered. In any particular server that's taking longer than that, in the long 5 percentile, then they retry to a different server with the same data. And that causes about a 5% increase of the load, but the new server will likely be very fast. But actually, it doesn't cost 5% increase because that it bounds the total latency to get all of the answers you need so much that you can run the individual servers hotter and you can get more throughput by playing this trick of retrying the really specifically slow ones. Fascinating discussion. Now, one of the observations I want to give to you is we can do this for writes also. And that's essential for tight service service level agreements like log writes to a database. So the writes now, in order to do that, they have to be idempotent. It's okay if I do them twice, and reorderable. It's okay if the writes land to the wrong place. And so they may land in funky times in different orders on different replicas. How are we going to make that work? Well, there are some tricks up our sleeve. First of all, I'd like to say 2 out of 3 ain't bad. So if I want to get an answer to at least 2 replicas, if I launch 3 and I wait for the first 2 to answer, then 2 records is durable enough that I think I'm not going to lose my log right and I'm in pretty good shape. So I launch 3, I get 1 back, and I say, cool, the log is now there. I can read it later. And the next trick is love the one you're with. Okay, you write to 3 replicas, but you want to make sure it's on at least 3 replicas. For example, no response from one, find others. If I have a large pool of places to write, I just don't wait very long to try somewhere else. So I write to 3, I only get 2, and so after a while I time out and I write to a 3rd. And now I've got it on 3 replicas. That's a trick I can use, but now the contents of each replica is kind of a mess, kind of tangled up. So I can bound the SLA by finding some place to write to, or maybe 3 replicas. I can make it fast, but I don't quite know where they landed. If I have some form of identity that can cope with that, then we can tolerate that confusion. The writes can arrive at the incorrect place, but it is okay. And it arrives at a replica different than planned, and I kind of have to shoo them home after a while. And of course, home is now something you have to be fuzzy about. If the writes on a particular replica arrive in the wrong order, I need to be able to know that and sort them out. So if I issue, buffer 1, 2, 3, 4, 5, 6, but different replicas see them as 4, 6, 2, 3, 5, 1, I may have different orders, but if the actual data has the order, then that can be cleaned up. And so I've now tolerated the latency by describing the place in a logical abstraction on top of it. And the intended order is assigned by the writer. It has to be intrinsic to what's written, and then each replica can reorder to its intended order. So assigning order at the client allows durable writes at any replica while preserving the logical order that the client assigned, even if they land at the replicas out of order. So tolerance of where you write can tighten the SLA for when you are durable. So I like to say Heisenberg was on the right track, W-R-I-T-E. In a distributed system, you can know where you write, or you can know when you write, but you can't know both. So we design systems nowadays for logging and other things to not care where you write, so we can know that the write happens in a timely fashion with extremely high probability. Now let's look at Schrödinger. So we've all heard of Schrödinger's cat, and I know it's not everything Schrödinger did, but it's what he's most famous for. He was a leading physicist in the early 20th century, tons of contributions to quantum theory, and he's most remembered for a thought experiment called Schrödinger's cat. In quantum physics, everything is in multiple states until it's observed, and that superposition of states collapses when it's observed. And so he thought that was pretty goofy. And he tried to talk and show how goofy it was by proposing that if you extend quantum level uncertainty to macro level uncertainty with the following experiments. You have uranium, a Geiger counter, cyanide, and a cat. You put them all in a box. And if the uranium decays, the cat dies. And if you believe this superposition of states, then the cat is both alive and dead until you look inside that box. So that was Schrödinger's thought experiment about a cat in the box. And so what's interesting is most physicists today believe that that's actually accurate. The cat is both dead and alive until it is observed. And that's a pretty interesting thing. And I want to take that idea and extend it to distributed systems. And I want to talk about what I call Schrodinger's put. So I'm working in a distributed system and I have these stores and they're like triple replicated or lots of other things, right? And you want to, some of them keep their data in unpredictable locations, which gives you prompt and timely SLAs. And you can relax where you write to tighten when you write, just as we discussed in the Heisenberg section. And here I'm talking about stuff that happens, for example, in the classic Dynamo from the SOSP 2007 paper, not the DynamoDB system that's popular now. If a client issues a put to 3 servers, if the system is rebalancing and moving around, the location of the write may vary. A subsequent get may have to look in many places. Similarly, if I'm writing to HDFS, you do what's called chain replication. You write to the primary, which writes to the secondary, which writes to the tertiary. And there are failure windows where you don't know how far that went, whether it's actually going to be there when you look. So if a client fails during the middle of that, we don't know much. The value that's being put may be anywhere or nowhere. If the get finds the new value, the software consciously rewrites it in enough places that before the guy who's reading it sees the get, gets that answer, the system is sure that subsequent readers of that, people doing get from that put, will get that same answer. And so while it is not yet observed, a put into a distributed system does not really exist. It's likely to exist, but you can't be sure. It actually simultaneously exists and doesn't exist. Only after being acknowledged or seen and rewritten did the put actually exist.
10 minutes.
Yeah, okay, we're good. Last slide is coming up. Okay, so this is the conclusion. It's really interesting to look at the perspectives. The conclusion is that we normally think of a Newtonian view of computing. As we think about computers as human beings, one clock moves forward consistently, and you can do transactions over relational databases. You can do a lot of things. And so in Newton's perspective, which is kind of medium scale, one clock, one now. Now, as you start getting into lots of computers spread out, you end up with distributed behavior, and the Einsteinian and Hubble behavior starts showing up. And as with Einstein, and this is also Lamport clocks, right? There's no consistent notion of time. Different paths may have different duration. And we see, similar to Hubble, there's an increasing pain over time as we wait for remote servers. In the cloud, Heisenberg and Schrodinger really confuse things. With Heisenberg, you know where you write, or you can know when you write, but not both. And the Schrodinger concept is that that change to the system, before you do it, it doesn't exist. And then there's both exists and doesn't exist. And then when you see it, it does exist. And so I'm done. Any questions, anything? Did any of that make sense to you guys? I see a bunch of moving blobs.
Thank you very much. That was a very interesting talk. I don't know how, other than moving shapes, can you actually see it? Anyone?
No, I cannot tell the individual blobs of human beings.
Yeah. So Pat, maybe you can comment on, so that Google was not discouraged by Eric's conjecture, and they still built Spanner, which is 1 million computers over the world using atomic clocks. So to what extent does the brute force violate, the consistency proof? So
Spanner does not violate CAP. Okay. It will stop availability of the data if it cannot communicate at a distance. It does guarantee consistency.
Okay.
There are cases when you're partitioned where you're stuck. You don't know what happened to that transaction. Now, Spanner is a really interesting and robust system, and it uses time to optimize a lot of things, but it can still get stuck if you can't talk across data centers. Now, it's less volatile because inside the data center, they tend to use Paxos, and Paxos can cope with servers failing. So it's more robust than typical.
Yeah, plus I think they also have atomic clocks and so on.
Well, the atomic clocks allow, it speeds the negotiation of how to advance the protocol. And so it's very useful to have the atomic clock. And I understand why they did it, although I think it's no longer needed given our ability to align clocks based upon what we've observed. But that's a long discussion. Any other questions?
Yeah, this is Charles Meneveau. A real interesting talk. Thank you so much. Seeing the word Schrödinger and cat on your slides, I was just wondering if you had any comments about quantum computing. How will that put that?
About what kind of computing?
Quantum computing.
I don't know much about quantum computing. I mean, I just don't. I'm fascinated. I would like to learn more, but I tend to be kind of an old guy who thinks of things in the normal ways, but I do not know exactly what that's going to be doing. I mean, of course, we all know that it's kind of like a non-deterministic finite state automata. If any of the paths find something, then you can answer that. But how the problems for which that's useful are not clear to me yet.
Thank you.
Pardon me?
No, you just said thank you. Any more questions? All right, if not, thank you very much once again. We had the next one.
Thank you very much for your time. Thanks for inviting me. I hope the rest of your conference goes well. Thanks.
Yeah, thanks, Pat. Come on.
So I do have a few more announcements before we all disperse. So first of all, thank you everyone for all the talks, excellent talks. This was an amazing day of very interesting talks across very diverse topics. So thanks again. I would like to thank all the people who have organized this, in particular, Elliot, Lawson, Kate Stevens, Biostatistics, and John Chou. Thank you. So one important announcement, if you have not had your parking garage ticket validated, please see Elliot at the desk before you leave. Unfortunately, so normally an IDIES tradition is that we have a cocktail hour at this point, but believe me, we tried, but the logistics just did not work out. So we're not going to have it this time, but at least we had an IDIES Symposium in person again. So that's a huge achievement. So once again, thank you. Thanks everyone. And it was just a great day.